Welcome to Download Day. My name is Tina Larson. I'm the President and Chief Operating Officer here at Recursion, and I could not be more thrilled to welcome all of you here to Salt Lake City to our event. And I'm going to, okay, yeah. So I'm going to kick it off and actually just tell you a little bit about my journey coming here to Recursion. I have been here for almost 5 years ago, almost exactly 5 years ago, when I first met our founder and CEO, Chris Gibson, who you'll hear from later today. And that was 5 years into the Recursion journey. So Recursion's going to hit our 10-year anniversary this year. And I got to join halfway through that journey. And at the time, I was based in San Francisco. I spent my entire career before now in the San Francisco area in the biotechnology industry. And I was working at a small biotech company, getting ready to launch a drug, launch a diagnostic, and certainly was not thinking about Recursion at the time. And when I met Chris and I started talking to him and I got to come here to Salt Lake City and see what was actually happening at this startup, I started to get really inspired and really intrigued. And the reason was because I believe biotech platform companies are the companies that transition and change our industry for the better. I'm a biochemical engineer by training. I spent the first 20 years of my career at Genentech. Genentech was the biotech platform company, certainly in the 90s when I joined, and really got to see, that company disrupt the industry. When I joined Genentech, they had no oncology products, they had no antibody products. There was still a lot of skepticism as to whether recombinant DNA technology and biologics were actually going to have a big impact on our industry, and obviously was able to see it play out, the huge impact that new technology had on bringing great medicines to patients. And so here we are, and I, 5 years later, could not be more excited than the day I joined about what now. Back then I thought maybe this could be the biotech platform company of the 2020s, and now I deeply believe it will be the biotech and is the biotech platform company of the modern era. So that's why I'm here. I hope you get to feel that while you're here today. What it's like now to work at the intersection of biotechnology and technology. And even at breakfast this morning, I got to talk to several of you, and it's so fun to see. Some of you are like, oh, I'm from tech, and then my colleague is from biotech, or I'm from biotech, and my colleague's from tech. And so it's really wonderful to see us bringing actually these kind of two different industries together. It's very much what we're about here at Recursion. So with that, I have the absolute pleasure of introducing our opening speaker today. And Martin Chavez is the chairperson of our board, and he's pretty intimidating to introduce. I'm going to be honest. I was actually just talking to him about this without taking like 25 minutes to tell you what he's done. But so I'm going to give a quick introduction, and I'm really excited we get to open with Martin because he has an incredible perspective and view of how technology is and can change the world. Both from the science and technology aspect, but also from the markets and financial aspects. So Martin had a very long career at Goldman Sachs. He was the CIO, the CFO, many other things over many years. He is also a scientist, a computer scientist, a medical information scientist, and he's worked with or been at institutions, Harvard, Stanford, MIT. He's also an entrepreneur, a former CEO, and he sits on so many boards. I couldn't even begin to remember all the boards. But importantly and impactfully, two of the boards he currently sits on are Alphabet and Recursion. And so with that, welcome up, Martin, and very excited to hear how technology is going to change the world.
Thank you. Thank you, Tina, for that wonderful introduction. I'm thrilled to be here. It's been a long road to get here. And so I'm going to tell you just a little bit about that, a little bit about the one area where I'm the dominant world expert, which is on me. And so we started off, I'm from Albuquerque, New Mexico, not so far away. And that's relevant because my very first job was at the time when the government decided that it was really bad to blow up bombs in Nevada and to test them. And started this huge, ambitious project. Could we just simulate the bombs and not actually blow them up? And that was a completely crazy idea in the late 70s, but it actually worked really well. And so I got to play on a Cray supercomputer, which is really dating myself, but that was very exciting for a 15-year-old in Albuquerque. When I got to Harvard, the first thing I had to do was declare a major, and all the science professors were recruiting for the departments. And so I go up to this table. There's a legend, I didn't know it at the time, Steve Harrison, sitting behind the table, was one of the people who first started crystallizing proteins to figure out how they fold. And he says, "What are you?" "What?" And I said, "I'm a computer scientist." And he said the following words to me in 1981, when I barely understood it. He said, "The future of the life sciences is computational." Imagine what that was like to hear that in 1981. And I thought, this guy must be right. And he said, "We'll create a major for you where you promise me to take one wet lab class. And then everything else can be any science that you want." And I thought that was a good deal, and it was very fortunate because I never got a single one of my electrophoresis experiments to work. Right? So it was terrible, terrible, terrible in the lab, but really good at writing software and building models and gathering data that was experimental and verifying it with the models and then going on from there. Now, I also had the privilege of working on crystallizing the tomato bushy stunt virus. You'll never forget a name like that, tomato bushy stunt virus. And this was in the Harrison lab. And Steve, even back then, said, "I am sure that if we know the amino acid sequence of a protein, and if we could just get the math and the software right, we could figure out how the protein would fold." This was in 1981. It took many, many, many years to figure this out, and you've all heard of AlphaFold, and I'm going to come back to AlphaFold because it's really, really incredibly important. Now, I then developed a fascination for artificial intelligence. I did a PhD in medical information sciences, but really AI, way back then, when we were embarrassed to say the words artificial intelligence because we could only handle really almost trivial toy problems. The combinatorics were so incredible. So I had, again, serendipitously, the chance to work with 3 other guys. So one of them is me, one of them is the chief science officer of Microsoft, one of them is Amazon's distinguished scientist in machine learning, and the 4th is by far the most successful. I shared a cubicle with him, and he thought, early on, medicine's too hard. Computers are not going to get there. Way too slow. They will get there, and he would tell me, but it's going to take another 30 years. I don't have time for that. So I'm going to use AI to solve a much simpler problem that people care about deeply, which is what movie to watch tonight. So that was my buddy Reed Hastings, who went off and started Netflix. And so the rest of us labored in obscurity for a long time. We felt really sorry for a bunch of people who were working on this thing called neural networks. I remember we would say, like that's going to work, right? And so we were completely wrong about that, been wrong about too many things and right about a tiny number of things. And so we kind of ignored them. And I really got lucky. In 1993, out of the blue, I got a letter from a headhunter, and it said, "I've been instructed to look for entrepreneurs in Silicon Valley with PhDs in computer science from Stanford and ship them over to Goldman Sachs." And I thought it was a joke, and I was very poor, and so I went on a lark, and I figured I'd scam this bank for a free trip, and then they put an offer in front of me, and I thought, okay, I'll work for a year and pay off my debts, and I stayed for 26 and ended up doing a bunch of things. So I had this very odd career path. People in Wall Street simply thought it was weird that I had gone to medical school. They couldn't make any sense of that. The PhD, that kind of made sense in the group that I was in, but it all seemed like a detour to me, and I spent most of my life thinking that I had wasted some time in that MD PhD program at Stanford and I should have gone straight to Wall Street. It didn't start to make sense until, really, I met Josh Wolf, who introduced me to Zavain, you're going to meet later, who introduced me to Chris Gibson. And Josh and Zav just said, we want to work with you on a company, and this company called Recursion is the one. And I learned a little bit about Recursion. So why am I so excited about Recursion? On the long list of things I'm doing, why is Recursion up there with my main gig, which is I'm at Sixth Street Partners, and my other little gig, which is that tech company that Tina mentioned, Alphabet. Why Recursion? And the reason is I figured out, like many of us, a long time ago, you can have really cool algorithms in machine learning. But if you don't have the data, it just doesn't matter. And so the thing that got me excited about Recursion when I was talking to Chris, he would tell me about the robots cultivating human cell lines and then systematically perturbing them in every possible way and gathering vast amounts of data and cleaning it and curating it and making sure that all the experiments are repeatable, reproducible, making sure the data is relatable. This is the sort of thing, it may seem like a geeky concept. It is so important. And you can have the best algorithms in the world, but if you cannot train it on reliable data, it doesn't matter. I'm in a bunch of industry conversations where everybody agrees that we need an immense data set of perturbation biology. I'm not going to jump the gun. You're going to hear a lot about that later in the day, and I am so excited about it. Why does this matter immensely? Let's go back to the protein folding example. Over 50 years, a bunch of grad students like me laboriously crystallized proteins. They documented their work in a standard file format. They committed it to the Protein Data Bank, which started in maybe 1971. AlphaFold is a magnificent piece of work. Without the Protein Data Bank, it would not exist, and you would never have heard of it. There was the data standards, the Protein Data Bank. There were competitions where there would be new proteins, and then people would see whose models best predicted the structure of those proteins before they were inserted into the Protein Data Bank. So about the time DeepMind, Demis Hassabis got to it, there were about 75,000 sequences, and these had been hand crystallized, every one of them, and laboriously curated. The miracle of AlphaFold was, it's amazing, training on those 75,000 proteins. Then, once the model got to a sufficient level of rigor, guessing the structure of 350,000 other proteins that were not in the Protein Data Bank. And this could have gone horribly wrong, right? The model could have trained itself and wandered into the weeds. But instead, it went incredibly right. Rapidly after that, predicted the sequences for 1 million proteins. And then not very long ago, AlphaFold inserted into a new data bank predictions for 200 million proteins, which as far as we know is every protein that exists in any life form on the planet. And now we're going to town on that. And while that is wonderful, and that is a miracle, and I remember people saying, chess, go, easy stuff compared to protein folding. But now protein folding is a solved problem. That is amazing. But the problem that we really want to solve is a much, much harder problem. And that problem is, I have a patient who's got some symptoms. And what I really want is for one of those Star Trek synthesizers to synthesize a medicine that I give to the patient, and then the patient is well again. Like, that's the dream. We are far away from that. That's the bad news. But here's the incredibly exciting news. I wanted to convey to you that I am an old-timer in AI. I've been around it for a long time. I've been through multiple nuclear winters. In the last 18 months, there is a miracle unfolding, and not enough people are paying attention to it. And the miracle is that whatever the AIs can do 3 months ago, now it's twice as good. And that has been happening for a while. So now we'll get to something else. You've all heard the proverb about the chessboard. The Shah wanted to reward the inventor of chess for creating this magnificent game. And the inventor said 1 grain of rice on the first square, 2 on the second, 4, 8, 16, 32, 64, 128, 256, 5, 12, 1024. I'm a computer scientist and all the powers of 2. And it's not very exciting. Now look at drug assets that are being discovered. 1, 2, 3. It looks pretty slow. It almost looks linear. There's nothing interesting about it. But then you start getting to the second half of the chessboard, and round about 40, you start to get to millions. And of course, by the time you get to the last square on the chessboard, there's more grains of rice than atoms in the universe. And that's the thing that's very hard for human beings to comprehend. Our brains just are not built for any kind of exponential compounding. And certainly not for the one that's been happening in AI for a long time. I think we've been doubling for a long time. It's just that the many, many doublings we've had have not been all that interesting. But now I can tell you from Recursion, from every other place that I'm in the conversations, the second half of the chessboard has arrived. And so while this problem of discovering the right drugs is immense, to put it in perspective, given compounds, small molecules of a certain size, there's, let's call it 10,000 trillion of them. 10,000 trillion. And right now, there's 4,000 approved medicines across the whole planet. 10,000 trillion to 4,000. That sounds daunting. And as I said, we're a long way from the solution. And we can't do this ChatGPT style. The other day I was playing with ChatGPT. It is amazing. I put something in, and remember I'm a computer geek, and it blithely responded, Python, unfortunately, lacks an addition operator. It just said that. I'm like, this makes my brain explode. I know I can type 2 + 2 into Python, right? So it makes stuff up that isn't true. And while that's cute on a chatbot, it is not cute when it comes to medicines, right? And so that's the thing that grounds us in reality here at Recursion. And what is happening here at Recursion is part of that miracle, that exponential compounding. We are in the second half of the chessboard, and unlike some of those chatbots, it is tethered to the most important reality I know, which is making people better. And so that's why I'm here, and we're going to tell you a little bit about it today. And turn it over to Chris.
Thank you so much, Martin. Really glad you all are here. I know some of you came from as far away as Europe, and it means a lot to us that you are here really as thought partners with Recursion. Before I dive in, I would love for us to show a brief timeline video, and during that, I'm going to grab the clicker. Awesome. That was a trip down memory lane for me. Over the last 10 years, we have built Recursion from a little tiny company with just a few people into what you see today. And I'm so excited that today you're going to get to experience Recursion the way we do. You're going to get to interact with our applications, you're going to get to meet our team, and you're going to get to see what we're building right up close. And I hope that throughout today, you're able to become familiar with our mission to decode biology to radically improve lives. And so I want to tell you just a little bit about the state of Recursion, the story of Recursion, and then I'm going to leave all of the exciting stuff to the rest of the team to share with you today. But before I do, I have something really, really important to share, which is this. We're going to be making small technical issue. We're going to be making forward-looking statements, and so it's important for all of you to understand the caveats that come along with that. So let me go back to almost day one. I think this was January 1st, 2014. This is a conference room at the University of Utah that we were able to sublease right next to a utility room, which we turned into our first lab. And this was the morning of January 1st, the first day our lease started. And I was sitting here starting to write our first grant, which was our first major funding source from the NIH. And we had one hypothesis. This was an experiment from the beginning. And that hypothesis was very simple. It was simply that in biology, structure suits function. And I'll tell you a little bit later today about the story of the first disease we worked on, even prior to Recursion, cerebral cavernous malformation in Dr. Dean Lee's laboratory. You'll hear a bit about that phase 2 trial that's now enrolling. But at that moment, we had this incredible, ambitious vision, which was to do an experiment to ask the question, could images of human cells be an unlocking event for this industry, a new kind of omics that would allow you to have an entirely different order of magnitude of data from which you could unlock not just a few understandings of molecular pathways, but you could start to actually unlock a map and navigate a map of biological and chemical relationships. That is how we began at Recursion. And if you go back and look at that grant, it's open sourced. It sounds like a very nerdy scientific version of what I just said. But over those first years, we stayed super scrappy. This is us in San Diego packing up a U-Haul full of used lab equipment, which we drove through the night, my co-founder Blake and I, back to Salt Lake City. We unpacked that lab equipment, and then we realized that we didn't have service contracts with any of the companies that had sold it to us, and most of it was useless. There was a ton of learning in those early days. But we stayed scrappy, and the data kept getting frustrating and then better, exciting, frustrating. But at the end of the day, it always came back to leading us to believe that this hypothesis of images being an unlocking event wasn't the top that we could get to. And we started to believe that there was something more ambitious that we could believe. And that's the second hypothesis, that if we leverage technology to build massive data sets, not just of images, but of transcriptomics, proteomics, metabolomics, in vivomics, all of these different layers of biology, we could turn drug discovery from this artisanal, bespoke, extraordinarily hard process into something much more like a search algorithm. And that's what we've been working on for the last 9 years, 2 months, and a few days. We've been working on that hypothesis. And today we're going to share with you the progress we're making against it. We started this mission at the right time. And that was as much vision as it was just dumb luck. I finished my PhD in Dean's lab on October 30th, I believe. We started the company on November 5th of 2013, which was at what I hope is close to the end of this 60-year trend in declining efficiency in our industry. And this is not for lack of effort. Incredible scientists have brought us miraculous medicines over this time. But the fact remains that biology, chemistry are so extraordinarily complex that it is getting harder and harder for us to bump our own knowledge as humans up against that complexity. And in fact, I would argue that today, we are bumping up against that line of human understanding amidst this system that is so complex constantly. And that's one of the reasons why it's getting harder and harder to discover new medicines. Along that same 60-year time frame, you look at the world of technology. Here looking at Moore's Law. You heard about Reed Hastings at Netflix. A little too early to go do the bio thing. We've seen the way that the exponential improvement in efficiency of technology has come to so many other industries. And yet we're just at the second half of the chessboard, as Martin shared, in the entire field, and it is now time to apply this to the field of medicine. And so Recursion was really founded to take advantage of this opportunity, this arbitrage that exists between declining efficiency butting up against this incredibly complex system, and the opportunity that exists with technology to actually charge full steam into that complexity. And that's what I'm so excited to be doing at Recursion, what I think all of us are so excited to be doing. So when you look at the traditional drug discovery process, it's incredibly laborious. Fantastic scientists are working to build new assays and new understandings for individual diseases, often sort of one at a time. They're developing a brand new assay at each of these steps in many cases to understand a different pathway or a different target in the context of a specific disease. But the reality is that the way we've built that system to date, the tools have not existed to make the data transferable across and between each of these boxes very, very fluidly. And what Recursion is trying to build is leveraging automation and machine learning to make data flow fluidly, not only across each of these boxes and between each of these boxes, but to make sure that the data generated in every program at Recursion can inform every other program we do. That the insights we're generating, even if they're too complex for humans to totally fully comprehend, are actually going to be available to that next scientist via a web application. And we still are doing all of this with the wet lab, as Martin shared, because that is essential to building the training set to enable us to start to map and navigate biology. And if we can be successful in this experiment, what the upside could look like is changing the shape of the biopharma funnel. Today, it looks pretty much like a V. Scientists do incredibly difficult experiments against many long odds to try and develop medicines that will make an impact for patients. And when they succeed, it's something to be celebrated. What we hope is that by doing this experiment, we can expand the neck of this funnel. By searching biology beyond what is in the literature, by finding relationships between chemistry and biology that one couldn't find just looking at the papers that exist today, and it would be very difficult to find using the traditional sort of unidimensional outputs. And then rather than just looking at efficacy early, we can start to measure things like transcriptomics and predictive ADMET and digital tolerability studies in animal models that enable us to not only look at efficacy signals, but to actually predict molecules that will fail early. Because what we want to do is narrow that funnel as quickly as possible and enrich our deep work as a company, as a team, in the programs that have the highest probability of success. And if we can do that successfully, and if we use robotics and software to do it at scale and at speed, then we can start to change the shape of this funnel into a T. And if you could get all the way to a T, that would be perfection. You could explore all of biology and chemistry, you could identify the perfect molecule and take it all the way to the market with no attrition. That would fundamentally reshape the economics of drug discovery, drug development, and our industry. And of course, that is unachievable, but we want to get as close to that as possible, and that's the experiment we're doing here. And these are leading indicators of our success. This is our funnel on the left in blue. You can see that since 2017, all of our programs are represented here, and you can see that we're starting to shape our funnel more like a T. There's more work to do, but we're making that progress. And what it means is for our programs, we can go from start to IND for much less and much faster than the traditional industry approach. And again, there's so much more work to do. We're in the very, very early days. I think these are leading indicators of what Recursion can deliver. So I want to tell you a little bit about 2022 before we set the table for all of the incredible insights and work you're going to hear from the team today. Looking back at 2022, Recursion matured this tech bio sector. We were one of the first companies to start clinical trials in this space. We were one of the first companies to sign some of the most significant partnerships in this space. And in 2022, we initiated 5 clinical trials. We're now planning a 6th clinical trial for our Axin-1 APC program. We have novel oncology programs coming down the pipe, including 2 I'm really excited about with Target Alpha, Target Gamma. Today, you'll actually get to hear we're going to unveil what Target Gamma is, a very exciting opportunity in the context of ovarian cancer. We're advancing some of the largest collaborations in tech-enabled drug discovery with Bayer in fibrosis and Roche Genentech in neuroscience in a single oncology indication. But what's important to remember, that Roche Genentech collaboration, that isn't just the largest collaboration in tech-enabled drug discovery. That is one of the largest blue sky collaborations to go for a decade across the whole of neuroscience to map and navigate neuroscience with Aviv Regev and her team at Genentech to try and deliver exciting new medicines against novel targets in some of the most important intractable diseases that face humanity today. That's a huge task, but it's one we're delighted to be working with our partners on. And today, you heard Martin say it. The dry lab by itself, not sufficient. Computation by itself, not sufficient. If you want to make a dent in this industry, you have to have both the wet lab, the dry lab, side by side, virtuous cycles of atoms and bits, making your algorithm better and better and leading you to better and better experiments. And at Recursion today, we have what we believe to be one of, if not the largest, proprietary, relatable biological data sets in the world at over 21 petabytes and growing, as you can see right this moment in the laboratory next to you. And you'll get tours a little bit later today. What this also means is that we are leading in an entirely new sector. When Recursion started, I think there were 10 companies that called themselves tech-enabled drug discovery companies. Today, there are hundreds, probably too many. Many of these companies focused on very, very limited point solutions. But if you look at some of the more advanced companies, our colleagues at Frost & Sullivan did this analysis, you can start to kind of look for the competitive advantages and differentiators of these companies. And you can see here on the x-axis, on the far right side, you can start to see companies who have focused on building that wet lab and that dry lab side by side. That's a major differentiator for these companies because you have to have the data on which to train the algorithm to make the kind of impact that we want to make here at Recursion. And on the y-axis, you see the maturity of these companies, the scale of the pipeline, the scale of the partnerships. Recursion in the upper right, leading this burgeoning sector. And I think for all of us who've been in this space, and even those of us who've been in large pharma or worked closely in that space, it now feels inevitable that technology will become a critical foundational underpinning of this industry. And it's a question of who, when, and how. And I think Recursion truly better positioned than almost anyone to deliver this value proposition to this industry. And that is important because we ultimately want to impact the lives of patients, their families, our team, and our community. So before I finish, I just want to share with you a little bit of a highlight of what's new today. We're going to give guidance for our pipeline. Two of the programs are phase 2 safety and efficacy study in CCM2, or in CCM. We're going to give guidance on our interim phase 2 safety readout for NF2. We're going to give guidance on our phase 1 C. diff program. We're going to give an update on our FAP phase 2 program. We're going to give guidance on the start of our phase 1b2 AXIN1 APC program moving to the clinic. And then also disclose the target for Project Gamma, which we're very excited about. On the partnership side, we're going to share for the very first time in our collaboration with Bayer a look into the pipeline that we're building together with them, and we're going to share updates on our partnership with Roche and Genentech, which has been extraordinarily exciting for this team. And finally, Martin alluded to this, we're going to share on the platform today, earlier this morning, we released the largest data set in this space. We released 2.2 million images of human cells across nearly the entire genome in an open-sourced fashion, along with what we call molecular recommender, or MolRec. And this is a tool at Recursion that we've been using recently internally to help us understand how small molecules are interacting with this complex space of biology. We've given people just a tiny snippet into this. And in the past, we've actually run competitions on some of these data sets. We've hired incredible talent off the back of some of these data sets and the innovative ideas that have come out of those competitions. And today, I think we're setting the table with the largest such data release of its kind to really continue to propel this field forward, not only at Recursion, but across the industry as well. And so ultimately, I want to end on our mission, to decode biology to radically improve lives. It's so meaningful to me. It's such a simple mission, but if we are successful in delivering this mission, we can have extraordinary impact on the world. And that is why every one of us at Recursion is here today, and I hope it's why all of you are here as well. So with that, I would like to turn it over to Tina to introduce the next team.
Can you stay for a second?
Are we going to banter?
Yeah, you did well. You did a great job, by the way. That was nice.
Can I leave?
No. Can I ask you a question?
Sure.
I like to banter with Chris, and you can tell it's his favorite thing. So, and I also don't like to plan for speeches. So, almost 10 years ago, 9 years and 2 months ago, did you ever think that that Recursion would produce more than 20 petabytes of data?
Yeah. It felt like we had a 1% chance of seeing this experiment through. And today it feels like that chance is much, much higher. It's not sure yet, but it always felt like this was possible. And if I'm super honest, I feel like we could be a couple years ahead, actually.
And that is Chris Gibson. Okay. Speaking as a COO. All right, so with that, I mentioned earlier that I love biotechnology platform companies, and I love technology, and so I'm very pleased to welcome up to the stage Imran Haque and Lina Nilsson, who are going to walk through our Recursion OS.
Hello, I'm going to test out this mic so I can use my hands. Hello, everyone. I'm Lina Nilsson. I'm the VP of Product here at Recursion. I've spent my entire career at the intersection of data science and human health. I have a background in computational biophysics and bioengineering. And I'm delighted to be here together with my colleague, Imran Haque, VP of Data Science, to share with all of you the story of the Recursion OS. What is the OS? It is the cumulative set of tools and processes that we have created in order to be able to build and interrogate massive data sets in biology and chemistry. It is the cumulative work of hundreds of Recursionauts over nearly the last decade. Or to harken back to the introductions by Chris, Tina, and Martin, it is a scientific and technical platform that we have built, fit for purpose, to be able to industrialize drug discovery, decode biology, and ultimately get more drugs to patients faster. Today, Imran and I are going to share the details about a bunch of really cool tools. But before we do that, I'm going to take a step back and tell you about the mindset behind the Recursion OS. And this is probably the most important slide in the whole presentation, because the mindset, that really is what underpins our strategy. It's what we believe makes us unique from all the other biotechs. It's the roadmap for how we will stay the leader in tech-enabled drug discovery today, over the next year, and into the coming decades. So let's dig in. In traditional approaches, literature tends to drive discovery. You start with a narrow, focused idea, maybe a target-specific hypothesis. If you're wrong, you've lost before you even started. In contrast with the Recursion OS, platforms drive discovery in an unbiased and target-agnostic way. The way we can do this is that we look at large swaths of biology all at once. As an example, using our phenomics platform, we have charted 3 trillion, so trillion with a T, biological and chemical relationships that our scientists can then mine for actionable insights. This unlocks transformative value for us as a company, right? Because it unshackles us from the literature, from all the things that the industry thinks are right, but maybe aren't so, right? The literature has lots of insights, but it's also riddled with mistakes, omissions, errors, groupthink. And we can move away from that by starting with our platforms. And not just platforms, maybe the key word is really data. In traditional approaches, data is often treated like an exhaust. It's the output of an hypothesis. In contrast with the Recursion OS, we turn this completely around. We use data as a fuel. In practice, what this means tactically is that our own in-house data is used for every hypothesis to drive every program forward. It is data as rocket fuel to ignite and then accelerate all of our programs. But not just any data, connected data. In traditional approaches, data is often siloed within individual programs and in time. They're single use. Imagine for a second 2 scientists working on a disease. They work in different laboratories, Maybe those laboratories are set at different temperatures. Maybe those scientists run the same experiment, but in slightly different ways. Maybe they get their reagents from different vendors. If you're just running that experiment twice, you can probably get away with that. But across many experiments, across large data sets, you lose the ability to compare the things you learned. And that really limits the value of that data. In contrast, at Recursion, our data is connected across our programs and across time because of the extreme focus of standardization that we have. I'll give you an example of the value unlocked by this. Let's think about the time dimension. If I look over here to my left, your right, you see the robots in the laboratory here churning out biological experiments and data right now, this minute. That data can be compared in a quantitative way with data we generated a year ago, even with data we generated 5 years ago, the year I joined the company. And that is incredibly transformatively powerful. And I'll give 3 examples of why. One is it increases our confidence, right? We don't just see that insight, we can look at the context of all the biology and chemistry around that insight. The second thing we get is we can discover the unexpected, right? We can look at data points that maybe we would never have included if we just limited ourselves to a starting hypothesis. And third, kind of zooming out a bit and thinking, connected data is an incredible competitive moat for us. And it's a moat that grows faster and faster over time. And the reason for this is the bigger your core data set is, the more value you get out of every single data point that you add, because the more connections that data set will provide into that core data set. It's a flywheel effect. And that's something that's really hard to catch up with for others. And to build to that idea of flywheel effects, I want to add a point here around traditional approaches and learning. There's, of course, learning in all drug discovery programs, but it's often siloed within programs, and maybe more importantly, maybe more nuanced, it's siloed between the physical experimentation as separate from the digital world, like Martin and Chris both talked about a little bit. In contrast, at Recursion, with the Recursion OS, we can create virtuous cycles of atoms, of physical experiments, and of bits of our machine learning algorithms. Fundamentally, the reason for this is because we're digitally native, right? We literally built the systems to be able to do this. I'll give an example into something. I'm really excited about the progress we're aiming for in 2023. There's a lot of new machine learning approaches that really require this interplay, like Martin mentioned, between the physical and the digital worlds. One example is new progress in active learning, a type of machine learning where iterative improvements in physical experimentation are done with improvements and iteration in your machine learning models. Physical experiment model, back and forth, iterating. For a company like Recursion, the value that can be unlocked if you do this well is that you get better at knowing what experiments to even run in the first place. So you're only executing the experiments that are the most likely to unlock actionable value. That's a really efficient and powerful way to leverage your research infrastructure and your resources. And if we zoom out a level here, really what this gives us overall is a way to really rapidly learn what works and then what doesn't, and to deploy changes not just locally, but across our whole research infrastructure at once, improving the company in big leaps all at once. And finally for this slide, and this is a summary of the slide, which makes it a summary of the whole presentation. If you compare a traditional approach, the focus there is on bespoke assays and processes. You work within individual programs, focusing on a single assay at a time that reveals a specific type of insight. That's really laborious, it's expensive, it's time-consuming. And in contrast, at Recursion, what we're doing is focusing on a more limited, high-power set of industrialized assay platforms. Things that can be automated and standardized so that we can run them day after day, year after year, to continuously generate insights across a whole portfolio. What this gets us in the end, the value unlocked, is that at Recursion, we don't have to approach our portfolio of drug discovery programs as a whole series of independent parallel efforts. Instead, we have a cumulative portfolio that we can push forward with one unified research infrastructure that's efficient, it's drug discovery, industrialized. I'm going to briefly show what this looks like superimposed to a traditional drug discovery pipeline, and Chris touched upon this briefly as well. All the way over on my right, your left, you have the initiation of a program, compound hit identification, all the way out to the beginning of preparing an investigational new drug application for the FDA. In a traditional process, this is very linear. It tends to be separate teams working on different parts, using different types of assays, with data stored in different ways. This really limits the amount of learning that can happen and the amount of process improvement, and it's in complete contrast to what we do at Recursion. For example, as I mentioned on the last slide, we deprioritized bespoke assays in favor of industrialized assay platforms. We start with phenomics, the unbiased study of the morphology of cells. We layered on transcriptomics, that's the high-dimensional study of the transcript, the step between genetics and proteins. And finally, even all the way out to in vivomics, the industrialized use of animal studies. You can see a lot of the other concepts from the last slide here as well, right? Virtuous cycles, connecting data, the idea of learning across the full portfolio. And ultimately what you get, if you do all these things well, and you do them repeatedly, like we do at Recursion, something really interesting emerges. And this is new. This has not been in the introductions or in my previous slide. This is what I'm maybe most excited about the work happening today. It's what I will work on when I get back to my computer after this presentation. It's what we call at Recursion industrialized workflows. It's the idea that we are not just automating individual assays, we're automating whole sections of the drug discovery pipeline so they can run in semi-autonomous ways. Imagine what we call an industrialized program generation workflow that can automatically generate insight packets that can be handed to scientists before they ever step up to the bench, before they ever pick up a pipette, before they put in any effort or time. And that packet can give insight about which compounds are the most promising to take into hit-to-lead activities. Another example, industrializing hit-to-lead and automating processes in the early optimization of chemical compounds. This is what bringing the recursion mindset to the drug discovery pipeline gets to, and I'm super excited to be here for it. So far, I've talked about the recursion mindset and how it's a competitive advantage. I'm going to turn to the other favorite competitive advantage at Recursion in my mind. It warms my heart to be able to share with all of you that data is also at the heart of Recursion. And we think about our data advantage across three big dimensions. One is scale, just simply the massive quantities of data that we're able to generate. A second is reliability, the quality with which we generate that data. And finally, relatability, our ability to connect that data to unlock novel, useful insights. And I'm going to give you three examples of this scale, reliability, and relatability as it applies to our data. An example from biology, an example from our robotics platform, and then finally an example from data science. First up, biology, 500 billion. That's the number of neurons we produced in 2022. That's a lot. We believe that might make us the largest producer of neurons on the planet, certainly one of the largest. If we were to commercially acquire this many neurons, we estimate it would be a financial investment of $500 million or so, assuming it's even possible to acquire this many neurons commercially this quickly. To take an example closer to me personally, I worked with human cells during my PhD, and if we had taken my approach, my protocols and throughput, it would have taken us thousands of years to get to 500 billion. So I'm glad we didn't do that. And maybe the most impressive thing to me personally about this slide is how hard it is to get to 500 billion. This is working with induced pluripotent stem cells. They have to be differentiated, expanded into neurons. This is notoriously hard to do under any circumstance. And we've had to develop the processes and the in-house quality control system to get to 500 billion cells in a way that's reliable and relatable enough that we could also build our new proprietary machine learning algorithms on top of it. And that's the real magic, I think. A second example, 2.2 million. That's the number of experiments we can run every week in this robotics laboratory right here. And the thing I want you to really key in on here is that it's 2.2 million, not a static number, that's 2.2 million flux. That's the amount that our competitive advantage grows every single week. Later on, during the tours of the building, you're going to have a chance to have a closer look in here through the windows and see the robots working. And when you do, I invite you to imagine for a moment all of that complex robotics infrastructure in an orchestrated dance with an equally complex digital infrastructure that you just can't as easily observe in order to execute those 2.2 million experiments, over 10 million associated microscopy images every single week. And what do we get from all of that? Like Chris mentioned, a massive proprietary data set of over 21 petabytes of data. I'm told that is more than all the feature-length movies ever made on the planet, in any language, in 1080p resolution, times 5. It's a lot of data. And like Martin mentioned, we all are appreciating today, right, the incredible central role of massive data sets in enabling modern machine learning. In internet applications, like in DALL-E and ChatGPT, we're all seeing the incredible outcomes that are underpinned by massive data sets. And that's exactly what we see also at Recursion, the insights we can draw from massive data sets. And maybe the big difference I would call out relative to many other applications, it's how high the bar is for biotech applications when you talk about the reliability and relatability needed. The reason for this is how complex biology is, how much of it is unknown by humankind. You can't have a control set so easily. And also, as a third point perhaps, just how important it is to get it right when you're talking about human health. And that's exactly why we at Recursion have invested so much effort over the last decade, not just to create a massive data set, but to create a data set that is reliable and relatable. So far, I've talked about data kind of generically, and I'm going to transition here to tell you how we turn our data into data layers and into maps of biology and chemistry. If you don't work at Recursion and you don't get to play with this data directly, this might be kind of hard concepts to wrap your mind around initially. Like, what's a map of biology? What do you mean a data layer? So I'm going to use an analogy to explain what this all means. And the analogy is mapping and navigating the physical world around us. Let's imagine you're standing at this intersection. You'd probably be able to tell me the number of houses there, what types of houses, the species of plants, perhaps. This is analogous to running one high-throughput assay in biology. You will get lots of reliable readouts, but they will be limited in the type, right? Let's go back to the intersection. Let's say I now asked you, where in the world are you? You probably wouldn't be able to tell me. You might be able to surmise it's somewhere tropical. I see some palm trees, but that's probably it. But let's zoom out a layer. Now, you would be able to tell me about the shape of the road grid, the topography of the terrain. And if you have been in this location before, if this is a known geography, you might even be able to tell me that this is on the outskirts of Honolulu, Hawaii. However, if this is somewhere novel, or novel biology, you might need more layers in order to be able to say with confidence where you are. If you're like me, you've never been to Hawaii, you might be able to need to see the whole state's island chain before you can say that with confidence. The take-home of this, through this analogy, is that we want to emphasize in on the power of being able to densely connect data within a data layer. Tell me about all the species of plants, but also the confidence and context you can build by moving between layers. Where in the world am I? And that's exactly what we're doing at Recursion by building lots of different data layers, lots of different maps of biology and chemistry. So for example, I've talked a little bit about our data layer in phenomics, in transcriptomics, all the way up to in vivomics and the study of animals. In fact, adding data layers like this, that's a core part of our strategy. We have ambitions to add layers in proteomics, eventually also in metabolomics, potentially, like Chris mentioned. I'm going to step out of this analogy, and I'm going to invite Imran up to give us a real-world example. We're going to zero in on the phenomics layer, our phenomics maps of biology and chemistry, and Imran is going to show us how that plays out with real-world data of both known and unknown biology, known and unknown chemistry, and how a drug discovery scientist might navigate through that to pull out some interesting learnings. Imran.
Thanks, Lina. I'm Imran Haque, VP of Data Science here at Recursion. By background, I'm a computer scientist by training, but my computer science friends call me a biologist. I spent about a decade in the biotech industry, in diagnostics for inherited disease, for cancer, and my biology friends call me a computer scientist. And I'm not exactly sure what to call myself, and that's why I'm here at Recursion, because I think that shared vision, that bringing together computer science and data, as well as biology and chemistry, is really key to unlocking the power of biology for human health. As Lina mentioned, what I'd like to do now is give you a more concrete view into how we apply our mapping and navigating technologies to actually discover drugs here at Recursion. You've seen some of the analogies and some of the principles that guide our data collection, collecting data at scale, collecting it reliably, and collecting it so that all those data points can be related to each other. Now let's see a worked out example. And let's do it with a disease that I think will be more familiar with, to the crowd, and that is breast cancer. So our maps of biology and chemistry are, broadly speaking, keyed by genes and keyed by compounds. So one of the things that a drug discovery scientist at Recursion might do is to start with a disease that's of interest to them, say breast cancer. You could then go to an external data set, say the Human Phenotype Ontology, and find all of the genes that are associated with the breast cancer. If you do that, you'll get the yellow edges that are listed in this graph up here, where you see a number of well-known breast cancer-associated genes, and then you start seeing the relationships that emerge in Recursion's map. Those are the green edges between those genes that are around the periphery. If you look closely at that list, you'll start to see a number of patterns emerge. For example, right here above breast cancer, you'll see a very tight cluster of the BRCA-associated genes. You'll probably have heard of genes like BRCA1, BRCA2, PALB2, RAD51, BARD1, all of which are strongly associated with breast cancer, and all of these show up as an extremely tight cluster in our data. Now, one thing that's critical to realize is that we didn't have to encode that into our systems. This all fell out naturally from the data that Recursion collects. We performed knockouts of each of these genes. We imaged those we imaged the cells with those knockouts on our microscopes, put them through our data pipelines, and these tight associations immediately came out. You'll see other clusters show up as well. So for example, another set of well-known cancer and breast cancer-associated pathways, RAS-RAF, HER2, PI3 kinase. Again, these are discoveries that took decades and hundreds or thousands of scientists to discover out there in the world that Recursion was able to immediately recapitulate from an automated analysis of its data. And we think that that's why the scale, reliability, and relatability of our data sets is so critical to what we're doing here. But if all we could do was look at genetics, this would be interesting, but not sufficient to transform drug discovery. And one of the really unique attributes of Recursion's platform is that we don't just see relationships within biology or relationships within chemistry, but we can actually see the relationships between the two. So let's zoom into one of these clusters to understand what I mean. And we'll zoom into this RAS-RAF, HER2, and PI3 kinase group. If we zoom into just that set of genes, what we can do is put up a set of well-known compounds to validate that the relationships that we're seeing in our platform are those that you would expect. So for example, at the bottom, you'll see a couple of well-known RAF inhibitor drugs, dabrafenib and carafenib, and you'll see that they have the relationships that you would expect to genes like BRAF. You'll also see other investigational stage molecules, AZD4547 and others at the top, and you'll see that they're making the relationships that you'd expect. These were originally developed as TGF-beta inhibitors, and you'll see that they have relationships, again, falling out automatically from our mapping and navigating data with genes in that pathway, like TGF-beta receptor 2, right there, right above breast cancer, FGFR1, and so on. So what the relationships on the last gene or on the last slide, and the relationships on this slide should do, is give you confidence that what our approach is doing is recovering real biology. We're able to recapitulate not only the history of genetic discovery in breast cancer, we're also able to recapitulate the history of drug discovery in breast cancer, or at least in this pathway in breast cancer. But if all we could do was recapitulate history, that wouldn't necessarily show the way forward. So you'll notice there are a lot of gaps in this diagram. Let me fill them in for you. What we have now is the rest of the compounds in the recursion data universe that show strong associations with the set of genes in this pathway, with these RAS-RAF genes. Now, I haven't labeled them because these aren't approved drugs, these aren't clinical stage drugs. These are starting points in Recursion's chemical library that we have screened that are also showing similar associations to these genes. So imagine for a moment that we lived in a world in which we hadn't done 25 years of research on the TGF-beta pathway, that we hadn't worked on EGFR and HER2. If we were in that world, we could take any of these starting points and then initiate research programs against them in order to drive towards drug discovery. And this is the real power of the Recursion platform. Not only do we recapitulate massive amounts of biology that we already know about, we discover new biology that was not known about or that was erroneously marked as not existing. We can then put that in relationship to large numbers of chemical starting points in order to initiate and advance drug discovery programs in the way that Chris and Lina have mentioned to you already. But this, of course, is just a small view of what exists in the Recursion data universe and in Recursion's maps of biology. This is zoomed in to about a dozen genes. So let's zoom out. Now, as Lina mentioned, we have over 3 trillion relationships in our maps of biology. This projector has maybe a few million pixels on it, so we're about a factor of 1 million away from even being able to represent all of these relationships. So this is a very schematic view of what you might see if you were able to zoom out into Recursion's maps. You see associations between diseases and genes in yellow. You see associations between genes, showing you how biology is put together in green, and you see relationships among genes and compounds in orange and purple, showing how all these pieces are put together in order to enable Recursion's scientists and Recursion's algorithms to navigate the space of biology and chemistry in order to move forward towards drug discovery programs. So in order to make that translation from maps to programs more concrete and show off some more of the technology that we've built to progress not just from relationships in the map, but really progress to that IND enabling phase, I'd like to invite Lina back on to go a layer deeper in the Recursion OS.
Thanks, Imran. So Imran just gave us a specific real-world example from within a map of biology in phenomics. You actually have 2 slides left. Do you want to come back up, Imran? Sure. He tells these 2 way better than I do.
That was a teaser. Now you get to learn a little bit more about the Recursion maps. So one point that I'd like to emphasize is what Lina mentioned about the way that data is treated as an exhaust in a traditional pharma setup versus the way that Recursion approaches data and approaches its direction to doing discovery. In a traditional target-based screen at a pharma, what you might do is look in the biological literature and identify what's the target that you would actually like to that you would like to go after in order to treat a particular disease. You'll then establish a unique experimental setup, a unique assay or assay platform to screen that particular target, and then perform a high-throughput screen against that one target in order to identify the chemical matter that you might then progress to hit to lead, lead optimization, and so on. Now, once you've done that, you've gotten a huge amount of information. You know, against that one target, what do all of those 1 million compounds look like? The problem is that's not nearly enough information in order to efficiently progress drug discovery downstream for a couple of reasons. From a scientific perspective, when you get that information about that one target, you're missing out on a vast amount of information against other targets. Polypharmacology is a well-known phenomenon in drug discovery and therapeutics today, where drugs don't have effects on just one target or one protein in the body. And in fact, the effects on multiple proteins may not just be important for safety in the sense that off-target effects might lead to toxicity, but it can actually be critical for efficacy as well, where modulation of multiple targets is actually necessary in order to drive efficacy against a particular disease or to drive differentiation of where different subtypes of disease may be affected by different drugs. Beyond the scientific reasons, there's also a critical financial or efficiency standpoint here as well. If you're interested in going after one target, great, you've got information about that one target. But when you want to stand up a new drug discovery program, you're now having to find that target, establish an entirely new assay, run the same screen again, and so the costs start to build up as you start to build that pipeline. That's not the kind of thing that you can do if you want to have that ultra-broad pipeline approach that Chris mentioned at the beginning. Recursion's approach is different. Because of the technology that we've built in our mapping and navigating approach, when we run a screen on our phenomics platform, each of those 1 million compounds that we screen can automatically get put into relationship with all of the gene knockouts that we have run, all of the compounds that we have run, all of the soluble factors that we've run. So what that means is that every time we run a screen, every compound automatically gets put in relationship to that entire library of gene knockouts, and we can model its activity against not just the one target, but actually all of the potential targets that we might be interested in. And that's the fundamental change that's able to turn drug discovery into a search problem, and in particular to turn it into an unbiased search problem, where not only do we find out if a compound was active against one target that a scientist may have been interested in, we can automatically find out whether or not it may have toxic effects, if it's showing up against other proteins that we don't want it to inhibit, or potentially if it affects multiple targets that may be important for the modulation of disease. So at this point, I think these are the two slides, and I will actually hand it back over to Lina.
So at this point, Imran and I are going to zoom back out and show how these different data layers, including the phenomics maps of biology and chemistry, are layered onto a drug discovery pipeline. I'm going to talk to you about the phenomics layer, give an example from transcriptomics, showcase a bit of our in vivomics work, and then I'm going to hand it back to Imran to show how we layer on chemistry on top of all of this work. So first up, phenomics. I'm going to give you a recipe for how we use our phenomic maps of biology to initiate drug discovery programs. First off, you have to actually physically have the experimental map. I would argue that the hardest part here is to model disease. Imran gave us an example using CRISPR-Cas9 to knock down genes across the entire genome. We also have lots of other perturbation technologies at Recursion that we build, we're currently working on, both in genetics and in other areas, like soluble factors or the use of toxins. Once you have your physical map, you need to capture a signal from that map. That's what happens here in the robotics platform, including in our microscopy stations, automated microscopy. And you're going to have a chance to dig into that later as part of the tours of the building. Next, you need to be able to detect and differentiate between cellular state, between healthy and deceased, using machine learning algorithms. Lots of companies say they do AI, and it can be hard to know if that's real or not. And I'm really excited to be able to share that at Recursion. You can go to rx.ai, you can download millions of images from this robotics laboratory, as Chris mentioned. You can download features that have been creating using our machine learning algorithms. You can look up papers and preprints and really dig into the way we work, dense nets, gradient reversal, the way we do batch normalization, and so on. So I invite you to do that and learn more about how we do machine learning at Recursion. But then finally, of course, having machine learning features, that's not the end goal. The end goal is to have insights that are useful for our drug discovery programs. And we do that in two main powerful and complementary ways. One, you heard Imran talk about. We create tools, web apps, that our scientists can use to mine that data. And later on today, you're going to have this chance to see a demo of another piece of software we use to also explore this data in order to extract actionable insights. The second way, I mentioned early in this talk, that's these industrialized workflows. That's the ability, for example, to find, verify, and validate insights from the map automatically without massive scientist intervention and effort. And one way we do this is to layer on transcriptomics. And I'm going to use this as my second example. Adding on transcriptomics in order to select which compounds we should put our effort in out of all those millions from the maps in our hit-to-lead activity, into chemical optimization, into ever more efficacious and safe compounds. The basic way this works is first we have a phenomic map of biology and chemistry. We run secondary phenomics experiments to verify the results. We then layer on transcriptomics, so an independent biological measurement, in order to ultimately get more confidence and more understanding about what compounds are the best to pursue and why. The transcriptomic layer here, traditionally, transcriptomics is often run sample by sample by sample. It's very expensive, it's time-consuming. And what we've done at Recursion is build a robotic version of transcriptomics using multi-well plates where we can look at lots of compounds at the same time. It's conceptually analogous to the phenomics laboratory right here. In fact, we just rolled out a dedicated transcriptomic laboratory space just across the road behind me here in the building next to us. And what we can do there is run some 15,000 exomes every week. And we actually have technical capacity to run much more should we decide to turn on the tap for more of those. Why is this valuable in practice? Why does the 15,000 matter? It means that we can create dense connected data layers also at the transcriptomic level. If we see something interesting in the maps of phenomics, we don't have to pick our one favorite compound or even our dozen favorite compounds. We can look at hundreds of compounds at the same time across many concentrations with lots of controls and compare it with lots of other biology of relevance. This means that we get both a nuanced and at the same time very broad net to help us decide what chemical matter is really the most likely to be successful at later stages. And with that, I'm going to transition to my third example of a biological data layer, in vivomics. In vivomics is the use of video in high temporal resolution animal temperature in order to run high-dimensional animal studies. And at the very surface, switching here to talk about animals and in vivo, that might sound like a big transition and break from the rest of this presentation, which has been based on the in vitro. But it's really the exact same story, right? Phenomics, that's the unbiased, high-dimensional readout of the observable characteristics of cells. In vivomics, that's the unbiased, high-dimensional readout of the observable characteristics of animals. It's also the recursion mindset showing up all over again. It's an industrialized platform. It's virtuous cycles. It's connected data. We're just now layering in the in vivo efficacy, tolerability, and liability insights. So let's quickly compare in vivomics to traditional approaches. Over here on my right is just one bespoke assay I could show you. It's a rotor rod study. These have been run in the 1950s. They're a popular way to measure the mobility of mice by putting them on these wheels and seeing how they do. In contrast, what we do at Recursion is run in vivomics over on the left. It's 24/7 capture of video of the behavior of mice in their original cages without handling and effects by human handlers in order to extract out in an unbiased way the signals that really differentiate, for example, the behavior of a healthy mouse and a diseased mouse in a way that is quite nuanced and doesn't require us to make assumptions about what signal is important and what isn't. It gives us a lot of value. One thing that's different is that we have continuous data generation. We don't have to wait for weeks to the end of a study to know if it worked or not. We can begin already on day one to begin to predict what's going to happen and what we should prepare for coming up. Many of the other benefits you see here are the exact same ones we saw for our other data layers. It's high-dimensional assays, they're unbiased in their readouts, we save time, we save money. All great stuff, but abstract, right? So let me give you some example what this actually means in our vivarium. So we have transitioned our entire vivarium, our animal facility, to be a digital vivarium, over 1,000 cages equipped with in vivomics. And we're going to give you 3 examples of the kind of studies that result. First up, in vivomic-enabled disease induction. Disease induction, that is ensuring that your animals really have the disease model you think they have, and then if they do, that you're able to detect that. That might sound like a trivial problem at first, but it's really not. Many diseases show up with much less than 100% population penetrance. That means not all the animals will get the disease. And second, even if they do, that disease signal can be really subtle. Lots of diseases cannot be readily studied using animal models. You don't quantifiably and repeatedly pick up on the signal. And this is exactly the problem that our in vivomic disease induction solves. What value does this give us? One, it lets us reduce the time, but quite a bit, in being able to set up new efficacy studies. And not on this slide, but what I'm really excited about, it also allows us to study diseases that have been previously thought not possible, not tractable using animal models. And now this whole disease space is open up for Recursion to pursue. My second example is digital tolerability. This is a method for figuring out which compound and at which concentration you should run in your efficacy study. Efficacy studies can be quite time-consuming and they can be expensive, so you want to be sure that you have exactly the right substrate that you're testing to get a high confidence on if this is a good path to go down or not. And that's exactly what our digital tolerability studies allow us to do, and importantly, they do that without delaying the onset of us being able to kick off that study. It doesn't slow us down at all. And then my final and last example here, liability in vivomics. When you run a compound in an animal, you test a compound in an animal, you obviously care to know that it works, right? That it's efficacious. But you also want to know that it's safe. And that's what liability in vivomics gets us. It allows us to compare the signature, the high-dimensional in vivomic readout of our compound that we're testing with known liabilities. For example, different types of liver toxicity or different types of kidney toxicity and so on. And that is also really powerful and something that we can do in a very efficient way. I've just now told you about 3 different biology layers, phenomics, transcriptomics, and in vivomics, and I'm going to hand it over to Imran to tell you about how we layer on chemistry on top of all of these processes.
So at this point, you've heard about the deep biology platform that Recursion has built, ranging from in vitro cellular manipulation and screening, with morphology, with transcriptomics, and in vivo data. The platform that we've built is incredibly flexible. We have chosen to do small molecule work on this as our primary aim for drug discovery, but we've also targeted this platform at a range of large molecules and biologics. We've done siRNA work, we've worked with proteins, we've worked with monoclonal antibodies. But chemistry is different, and the way that I'd like to communicate how different chemistry is, I shared what computer science friends and biology friends think of me. Despite literally having done a PhD in computational chemistry and cheminformatics, not once have the chemists called me a chemist. And in order to show the ways that Recursion is addressing those high standards that chemists and pharmacologists hold themselves to and hold their products to as their molecules move forward into the clinic, I'd like to share some of the work that we have done and that we aim to do in advancing chemistry capabilities in the Recursion OS. Our vision is, just as we've done in biology, to integrate both digital and robotic operations in order to advance chemistry, in order to try to reduce our time to candidate selection from months to weeks. Now, you may be familiar with a notion in medicinal chemistry and drug discovery known as the design, make, test, analyze cycle. First, a chemist or an algorithm will design a molecule. That molecule has to actually be made or ordered from a catalog. You'll then test that in a suite of different assays, whether they're biological assays like those that we run on our platform here, or other assays that you might need to run for ADME tox or DMPK reasons. Analyze all of that data, and then design the next round of molecules to go through that process, hopefully increasing your understanding and the quality of the molecules at each step. Our approach is to develop technology in each one of the phases of the DMTA cycle in order to improve its efficiency in 3 different ways. First, to reduce the number of cycles that we have to make around this loop in order to reach the molecule that will be a clinical candidate, or that will be a development candidate, or to terminate the program. Second, to shrink the time required for each time around this cycle. Time is critical. We would like to prosecute a broad portfolio of drug discovery, and speeding that up makes a difference. And finally, to increase the throughput at each around the cycle, to be able to test more molecules at each step in order to maximize the chances that the programs that we advance and the molecules that we advance are the right ones for the patients downstream. So let's talk about the ways that we are making advances in each of these steps, starting with the design and analyze processes in our digital chemistry platform. We've talked a lot about the things that we have put into production here at Recursion, and I'd like to highlight a little bit of the research contribution that we've made as well. In particular, a pair of papers that were presented at workshops at NeurIPS 2022 in December, arguably the premier conference in machine learning, show off the work that Recursion's research scientists have done to push the bounds of research and the bounds of advancement in chemistry and chemical machine learning. One paper on what are called multi-objective G-flow nets discuss our advances in generative chemistry, how we can have algorithms design new molecules, select new molecules to enter this DMTA process. As I mentioned earlier when talking about the map, drug discovery is intrinsically a multi-objective problem. There's not just one target that you care about, there may be multiple targets, and then you also have to think about considerations like the pharmacokinetics or the metabolism of this drug. Generative chemistry algorithms, therefore, need to be able to optimize multiple parameters. And the algorithm devised by Recursion scientists, multi-objective G-flow nets, outperform other benchmarked algorithms in numerous multi-objective settings in chemistry. On the analyze front, we've built a machine learning model that we call MOLE, a molecular foundation model for drug discovery. A challenge in drug discovery, as compared to other domains of machine learning, is that the amount of data that you have is actually quite small. We talk about 21 petabytes of data, but when you're at the beginning of a cycle for optimizing a set of compounds for a particular drug, you may only have a few dozen or a few hundred data points. That's not that many to go off of. And so what you need are machine learning models that can learn from history and then very efficiently learn to predict properties on the local series that you have for a particular problem. What our scientists have done is adapted methodology and technologies from large language models that power things like ChatGPT in order to train what we call a few-shot learner in chemistry. And this model achieves top 1 or top 2 performance on about 2/3 of the ADMET tasks in a public benchmark dataset called the Therapeutic Data Commons, including all of the distribution and metabolism tasks, and is the top performer in 9 out of those 22. We've already started to use MOLE and G-flow nets to power the internal drug discovery processes and the virtuous cycles in our programs. Now, beyond the contributions that we're making on the digital front, we're also building advances on the physical and the robotic front of chemistry. Behind you and to your right, we have our brand new automated DMPK testing facility, a Recursion proprietary design to automate the execution of 3 critical DMPK assays on hundreds of molecules per week. That's enough bandwidth to not only get the data that we need on the programs that on the molecules we're progressing for our programs, but also to provide additional data to fuel our machine learning, to turn these traditionally extremely difficult tasks in ADME and tox into predictable problems that we can solve using our models. And finally, on the make front, Recursion has ambitions to build automated microsynthesis systems, where we believe that automation of chemical synthesis will enable broader SAR cycles, broaden the number of molecules we can test at each point in the process, and enable a faster turnaround time for active learning cycles. I want to return to that concept of active learning that Lina mentioned. The notion here is that you can have a computer algorithm pick what are the next places in space, in chemical space, the molecules that you might test, in order to not only optimize the molecules, but optimize the model's understanding of the problem in order to pick the best molecules downstream. Now, as I mentioned, drug discovery is an intrinsically multi-objective problem. Active learning has been of interest in the pharma industry for literally decades. But I believe that the multi-layered data that we're building here at Recursion doesn't just empower drug discovery scientists, it empowers our algorithms. And our goal is to automate and industrialize as many steps in this process as possible. And the breadth of data that we collect is truly key to enabling that, because you can only optimize those pieces that you see. That's why we build scalable, reliable, and relatable data sets across these multiple layers of biology, across the morphology and the transcript levels of individual cells, moving up to the DMPK properties of molecules, and indeed the entire organism-level properties that we assess in vivomics. So with that, I'd like to take this now to one more level of concreteness and introduce my colleague Laura Schaevitz, PhD, SVP and Head of Research at Recursion, to discuss some of the opportunities that we see in our preclinical research.
A little shorter here. Can everyone hear me? Perfect. Hi, I'm Laura Schaevitz, SVP, Head of Research here at Recursion. A little bit on my background. I'm actually a PhD neuroscience from Stanford University. And before coming to Recursion, I was actually the Chief Scientific Officer and then CEO of a company called Vium, which built the technology underlying the vivomics that Lina and Imran just told you about. And so one of the things I just kind of want to start with here is when Recursion offered to acquire Vium 2.5 years ago now, I think it was, I was really excited. So the technology we were building at the time, we were trying to get adopted into the mid and large pharma companies, and adoption was maybe a little bit slower than we would have liked. And I think a challenge there is just finding companies with the right mindset. And Recursion is that place. I think looking at what Vium was trying to build and what Recursion is trying to build, there was just a beautiful mesh. And so what I'm really excited about sharing at the end of the presentation here is how we're actually employing that technology on our programs today. But before we get there, I'm looking forward to sharing with you how we're using the Recursion OS to both discover and drive some of our internal programs. So to start, I'm going to give you a little different view of our maps of biology than what you saw before. So this is the heat map version. And just to give you just a little bit of an orientation, what you see on the, let me see if I get this right, what you see on the rows and the columns here are the individual genes, and we're looking at relationships across these maps. And so along that diagonal, you see self-similars, they look great. What you also see are these clusters of biology, where what we're seeing are those genetic perturbations that look very similar to one another. By taking this really, and what I guess what I would also say is this is just such a tiny snippet, as both Chris and Imran and others mentioned. Our actual maps contain the whole genome knocked out and hundreds of thousands of compounds. And by taking such an expansive approach to how we look at biology, we can really use these maps in very different ways to uncover kind of those novel insights in programs. And I'll walk through a couple of programs today with you. But just to give you like an overall sense of what that might look like, we can use our maps to uncover what was potentially considered at one point an undruggable target. So for example, if we put that target into our map of biology, we can look for other genes that look phenosimilar to it, which would give you a sense for maybe these genes act upstream, downstream, or even potentially adjacent, but still provide that same phenotypic output. The other areas that we can potentially target, let's see if I get this right, are adding in some of our patient genomics, the large patient genomics data sets that are out in the world. We can combine those with our maps to really uncover those interesting novel insights. So let's actually dig into a couple of our programs. So the first program I'm going to tell you about today is Target Alpha. And for Target Alpha, we're combining these external data sets with our maps to find what we think is a potential first-in-class NCE with novel, we say MOA here, but the reality is this is a compound with polypharmacology where both of the activities of the compound we think are really essential to be driving this enhancement to immunotherapy. So we all know the story of immunotherapy. We all have probably heard 1,000 pitches for immunotherapy drugs or combinations with immunotherapy, and I'm going to tell you that this one is a little bit different. So in traditional immunotherapy, we know that of the 44% of patients that are eligible, a huge fraction of those patients don't actually respond to immunotherapy alone, upwards of 88%. We know that of those who could potentially respond, a large percentage of those patients have these immune-related adverse events which prevent them from actually being able to take the drug that could be effective for them. And so at Recursion, we're ambitious, and our goal was not just to find another immunotherapy or another drug that could be used in combination with immunotherapy, but to be able to find a drug that could both increase the number of patients that would respond to immunotherapy drugs and also minimize those peripheral side effects. And I think we might have something. So let me tell you how we used our maps to find the starting point for this program. So our oncology team took a couple of very famous papers, Manguso and Larson, which describe about, I think, a couple thousand causal markers of either enhanced or resistance to checkpoint immunotherapy. They pored over them. They narrowed it down to about 110 genes that they were particularly interested in for a number of reasons, including perhaps which ones show nice phenotypes in our maps. They mapped them, and then they looked for interesting relationships. And so let's actually walk through the map. So here is the actual map of all 110 genes. You see the nice self-similars, and you also can see a lot of very nice clusters of these genes. And one of the clusters that we were most excited about is a cluster that is right up in here that contains a gene called BRCA2. And if we zoom in on that cluster, you can nicely see BRCA2. You can see BRCA2 with a lot of other BRCA2 family members, gives us great confidence. And then there's this extra gene called gene A, and I won't tell you what that is. But gene A is a known immune modulator. So that was kind of interesting. So we took that gene and we put it back in those broad maps. So all genetic perturbations, all of the chemistry we had, or all of the compounds we had at our disposal. And we found two interesting relationships from that. The first interesting relationship was the gene A we found an actual inhibitor to gene A, which is always good when you can find that. But we also found a second gene, gene B, that gene A and the inhibitor to gene A seem to be phenosimilar to. And this one was really exciting because gene B turned out to be an up-and-coming resistance marker for immunotherapy. So now we have potentially a compound that is phenosimilar both to gene A, which is immune modulator, and gene B, which is a resistance marker. And we actually went ahead and we confirmed that this compound was active against both of those targets. So now we're getting pretty excited about this program. We potentially have a compound with polypharmacology that can be hitting kind of multiple different levels of this program. So we went ahead and put it into an animal model. And for this program, we used a CT26 animal model. And we use that because it's fairly highly resistant to immunotherapy. And as you expect, I'm a pointer, so as you expect, here for the vehicle and the anti-PD-1, you see really no effect on tumor volume. But now we're starting to get excited because we see a monotherapy effect for our compound with an actual improvement when given in combination with anti-PD-1. Upwards of, actually for this combination, we saw upwards of 40% complete responses. When we took those 40% complete responders, we re-challenged them. We saw that 3 of the 4 had no tumor regrowth, suggesting immunological memory. So this is looking pretty good. We then looked within the actual tumor microenvironment to see what was potentially going on there. We saw an increase in immune infiltrates, as evidenced by these total CD45 positive in the combination. We also saw decreases in suppressive macrophages, as well as LAG-3 exhausted T cells. So generally looking very good within the tumor microenvironment. We also looked at cytokine levels, and I'm showing you here just 2 examples of course, a panel of cytokines that we looked at. Within the tumor, evidence of immune response within, or I would say similar immune response across all of our treatments. But remember, our goal was not just to improve response in patients in combination with immunotherapy, it was also to decrease that peripheral inflammation. And so we got really excited when we looked at cytokine levels in the periphery. And so here, you can see plasma levels of these same cytokines with the anti-PD-1 elevated levels, which are significantly reduced in the combination. So now we potentially have a compound which is showing efficacy in our animal model in combination, and when we give our drug, we're reducing this peripheral systemic inflammation that's potentially limiting the ability of patients to be able to take these, to be able to respond to immune checkpoint therapy. But of course, the compound is not ready to go into the clinic. There's work to be done. We need to optimize it against both of our targets, and we needed to do some work on drug exposure. So our amazing medicinal chemistry team actually turned to the Recursion OS in order to help them drive chemistry optimization. And now you're going to get to the geeky part of me, because I love where the technology and the biology actually intersect with one another. So for this program, we had all of our compounds synthesized in an external CRO. At the CRO, we knew we were trying to optimize against 2 different targets. So we set up, biochemical assays there, as well as reporter assays. But then we also had the compound shipped to Recursion, and we immediately profiled them on the map. And what we wanted to see above me here is the compounds all looked similar to one another, and that they looked, that their phenotype looked similar to that target phenotype that we really cared about, which should represent the polypharmacology that we cared about for this program. Now, the map helped us stay really laser focused on this target phenotype that we cared about, and I'll show you an example of how it did that. About halfway through the program, our chemists introduced a second core, and you can actually see here that in this new core, our reporter assays started reporting out that potentially we had increased cell-based assay activity that was potentially really exciting for us. Now, possibly in some other place, we would have been excited about this, this new core. But at Recursion, we never got excited about this core. I think the excitement lasted for all of maybe 5 days, because as soon as it was profiled on the map, if you look above me, you can see these 2 new compounds here, but their phenotype was not at all like the target phenotype that we cared about. And so we knew that we had done something in introducing this new core that was not something that we cared about. And of course, we're interested and excited chemists and biologists, so we wanted to understand what it was exactly that we had done. And this is like the true power of our map. We didn't have to run an experiment. All we did was we took these two new compounds, we searched them in our broad maps of biology and chemistry, and we said, is there a gene knockout that looks somewhat similar to this? And lo and behold, there was. So here we are. And that gene was CDK9. And we actually went and confirmed in a simple biochemical assay that those compounds were active and inhibited CDK9. So a beautiful example of how we can not just drive the chemistry, but if something goes off the rails, we can understand what it is that we did without having to assess or set up a whole bunch of new biochemical assays to be able to do it. So let me just give you the kind of overview of target alpha, and then I'll move on to gamma. So I showed you here how we used our maps to combine external data sets with our internal map in order to find this magical compound that has polypharmacology that can potentially both increase response to checkpoint inhibition as well as to decrease peripheral inflammation, and then how we used our maps to actually augment our chemists to drive chemical optimization for this program, including when we went off the rails, what exactly had happened. So now let me turn to target gamma. And in this transition, I want you to remember, and I think this plays back to a lot of what Lina and Imran were talking about, around how we try to use exactly the same strategies over and over again to industrialize these processes. I'm kind of going to tell you exactly the same story for gamma, so please don't go to sleep on me. But I'll try to make it a little bit more brief this time around. So first, we introduced target gamma 15 months ago, and I'm actually excited as we're doing this to announce to you what the actual target for gamma is. And target gamma, we are going after an RBM39 degrader for this program. And let me give you just kind of a high level of the philosophy of the program, and then I'll get into how we used our maps to actually find RBM39 as this potential target for gamma. So the philosophy for the gamma program was the following. We were looking for a molecule to be given in combination with PARP inhibition for patients who are homologous recombination proficient, which is a large patient population that currently has no viable treatment options. So for ovarian cancer, first-line treatment, as many of you already know, is debulking platinum-based chemotherapy. Many patients respond very well to that and see complete tumor regression. If you give a PARP inhibitor, they're a large portion of the patient population, 50% that are homologous recombination deficient will actually respond very well to that PARP inhibitor, and you'll see an enhancement in progression-free survival for those patient population. But if you look at the 50% of the patient population that are HR proficient, you see almost no increase in progression-free survival for that population. And so this is the population of patients we were really going after for this program. So now, hopefully, it's the part you have all been waiting for. How did we identify RBM39 as this alternative target for CDK12? So we started with CDK12 because it's been known to be a target to improve response in the HR proficient setting. But we also know that it is a challenging target to go after because it is highly homologous to CDK13, which has a number of toxic side effects. And so our goal here was to be able to find a target which was adjacent to CDK12, We don't know exactly how RBM39, is it upstream, downstream, is it a side, or is it adjacent with that same final phenotype? But that's what we were looking for this program. When we turn to the map, getting again to a bit of what Lina and Imran were talking about around biology may not actually always look like what you expect, we actually found that CDK12 and CDK13 are not at all like one another in the map. And so that was kind of an interesting finding on its own. Then when you look, when you put CDK within that kind of broad expanse of our map, we found that CDK12 was very phenosimilar to RBM39, which is not known or documented at all in the literature, and that we also found this compound REC65029, which is highly phenosimilar to both of those. We did confirm that our REC65029 is in fact acts on RBM39 and does not act at all on CDK12 or CDK13, so it is very specific to RBM39. In this program, I'm not actually going to show you at all how we do it in the interest of time, but I will tell you that unlike Target Alpha, where we set up biochemical and reporter assays, we actually drove SAR for this program, or chemical optimization for this program, 100% using our phenomics maps. That was the only tool or platform that we used in this process, which I think is really amazing and impressive. I totally skipped ahead because I was so excited about that part. My apologies. Let me first get to the important piece, which you probably all care about, which is the animal data. One of those little hiccups, right? So for this program, we put this molecule in combination with PARP inhibition into an HR proficient ovarian cancer model. As you expect in this model, PARP inhibition alone has no effect. Please notice that that is a log2 tumor volume scale. We were really excited to see that our compound alone showed very significant effects. And when you give it in combination with the PARP inhibitor aliparab, you can see even increased efficacy in that model. If you look over here on this side, you can actually see very significant or faster rate to complete responses in the combination versus the single agent alone, and a durability of the response in the combination versus the single agent alone. But it is exciting that we saw single agent efficacy for this model, or for this molecule as well. So now to the piece that I was excited about, the fact that we were able to really use the platform to drive SAR alone, I think is really significant for us at Recursion. But now I want to take you, so we did some work, we optimized the molecule. In this case, we were looking for a molecule that had a better therapeutic index than this starting molecule. And let me take you to the piece that I'm potentially really excited about because I'm biased. And this was the work that I did for 5 years. And that is how we used the InVivomics platform to help us pick and figure out what molecules and at what doses we should take into efficacy studies and potentially into the clinic. So again, here is the beautiful images of the animals in the cages. And as Lina was referring to earlier, we take 24/7 video and we automatically read out behavior from the animals. And what I'm showing you up in this corner here is that first compound that we identified, REC65029, and the actual profile for that. This is activity measurements. And as many of you know, animals are nocturnal, so you see these beautiful circadian rhythms with high levels of activity at night, low levels of activity during the day. For this molecule, in black are our vehicle-treated animals. You see some depression of activity in some of the higher doses, really speaking to the need to get to potentially a better therapeutic index than what this molecule could provide. As we ran through our chemistry optimization, we identified a number of molecules that were potentially quite interesting in terms of having the drug exposure properties that we were looking for, the potency that we were looking for. And here are just two examples, and I think this is a really good indication of how we can use technology like this to help us move faster. So the first molecule I'll draw your attention to is this Rec 11700204, in which you can see, again, vehicle looks fairly normal, but at both doses that we gave, which were required for efficacy, we saw very significant decreases in the activity levels of these animals, suggesting potentially some safety concerns for us. Whereas in this molecule, Rec 1321245, you can see little or no changes in the activity levels at all for these animals at doses that were required for efficacy. And so this really is an example of how we can quickly assess the potential safety of a molecule in an exploratory study. And this really helped us hone in on this is probably the more kind of safer, efficacious compound to move forward. And what I would say for all of these is that we have filed a number of patents around this, both around method of use as well as a number of these molecules in terms of composition of matter. So let me kind of end with just a reiteration for the target gamma program. While I didn't show it, I showed you how we identified kind of that the relationship between the novel relationship between CDK12 and RBM39 using our maps. I told you a bit, but I didn't show you how we used our maps and our phenomics to really drive SAR for this program, and then how we're starting to incorporate some of these other high throughput assays that we're working on to help us figure out what are the right molecules that we need to be driving forward as quickly as possible into the clinic. So with that, let me give you the quick where are we going in 2023 for our preclinical programs. We are working to drive target alpha and gamma to IND-enabling studies in 2023. We're continuing to augment and bring into our internal programs digital chemistry and predictive capabilities, which Imran started to touch upon, but I wasn't able to show you any really good examples here today, but hopefully the next time we will. And then the areas that I'm really excited about over the last couple of years is we've seen how we can use our platform to really drive these programs in highly consistent ways. Gamma is a perfect example. We're no longer setting up bespoke assays to be able to identify and drive our programs. And so really in 2023, we are doubling down, and I love being partners with everyone here, including Lina, on really setting up these industrialized workflows for our preclinical programs, such that as much as possible, we're taking the scientists, I wouldn't say totally out of the loop, but we're taking them a bit out of the loop. It's sometimes uncomfortable for them, but we're taking them out of the loop on some of these early stages of inference and hit to lead, so we can drive programs faster and at scale to really deliver on the mission of what we have here at Recursion, which is to decode biology to radically improve lives. And with that, I think, thank you, and turn it over to Tina.
I lost my mic. All right, thank you, Laura. And so Chris mentioned earlier, what are the new things we're going to learn today? So we just learned RBM39 is target alpha, and I'm going to invite Ben Mabey, our Chief Technology Officer, up here to tell us more about what Chris also mentioned, the release of a data set and app today. And then we're going to head to tours, so almost there.
Thanks, Tina. So I'm the CTO here at Recursion. My background's in software engineering and machine learning. I'm also a proud father of 3 kids who love to build things. The kitchen table's always a mess full of cardboard boxes and other craft building materials. and their rooms, it's like a landmine of Lego bricks, quite painful. So on Christmas morning, I gave them a 3D printer, and the reaction was absolutely epic. They were so excited for it, jumping up and down, dancing, and it's been a joy watching them for this past month to take their creativity to the next level. As they're like, the world of possibilities has really been expanded with this new technology. Now, as we get older, that childlike curiosity tends to dissipate. We're less excitable. But I see that same reaction when I talk to new scientists and computational biologists at Recursion. I ask them, why did you join? And it's always, they talk about things like this map, all these examples you've been seeing today. They get so excited about that. And I can say that that excitement is contagious beyond Recursion as well. A few months ago, I was in a meeting with some of our partners, and we were talking about a set of genes, and one of my, a director of data science DM'd me. He said, hey, Ben, can I show them the map here? I'm like, sure, go ahead. So in 20 seconds, he shared the screen, pulled it up, and we were looking at the relationships, and their jaws just dropped. It was so fun. It was like they had been given a 3D printer. And so this, the idea of this excitement, that's one of the motivating factors behind this dataset release, which Chris mentioned. We want to share with the world a sliver of that excitement so people can understand the future that we've been building here at Recursion. So Martin alluded to a database of perturbations, and that's what RxRx3 is. It's a map of phenomic biology, and it spans the entire human genome, roughly 17,000 genes. It also has 1,600 compounds, and these compounds are FDA approved, commercially available, and we've screened them at multiple doses. All in all, there's 2.2 million images, so 2.2 perturbations, and they're associated deep learning embeddings, all in the cell context of HUVEC. And this clocks in over 100 terabytes of raw image data. Now, that 2.2 number should sound familiar to you, because that's the number of experiments Lina mentioned we're able to run in a single week here at Recursion. So while objectively this is a large data set, it represents merely a week of capacity. Chris also mentioned that we've done this before. We've had multiple data set releases. This is our 5th one. And we keep doing them because we like to give to the community, but it's also good karma. We have received dividends from these releases, such as being able to hire people who have used the data sets in publications, in industry, and academics alike. Now, while we've done it before, we've never gone this big. This is a data set that is 76 times larger than any of our other previous data sets combined. So this, as Chris mentioned, we believe this is the largest data set of its kind to be released in this space, and it is, without a doubt, the largest, most highest quality data set because it's been able to be produced in one location. It's hard to find that kind of data in industry, let alone public domain. But remember, this is coming from the Recursion data universe, which is 21 petabytes. And so it's actually less than 1% of the phenomics data that we have, which spans roughly 50 human cell types. Now, the majority of this data is gene knockout data, as I mentioned, but there's a small component of compound-specific data. And it's that compound-specific data that we're going to allow people to explore using in an application. Just like I mentioned that story of our partners that their minds were blown when they saw the app. There's something magical about seeing the data in use. Because it's one thing to know you have petabytes of data, but once you can navigate it quickly and efficiently, that's when the lights come on for many people. And so we want to have that experience with people outside of Recursion, and so we're releasing MolRec. You can think of MolRec as a demo application, similar to how Apple might release a demo application highlighting new capabilities of a new iPhone. We're releasing this application to highlight what can be done with our data. To be clear, it's not our flagship internal map application. You're going to be able to see that later today in demos. But this is more of a simple version just to give people a taste of what's possible. And so with it, they can explore from a compound view, compound-compound relationships and compound-gene relationships. And people familiar in the drug discovery industry, they'll feel at home in this app. There's terms and analysis and plots that are very familiar. Cytotoxicity, EC50s, and concentration response curves. What's different here, though, is that, like we've said all this morning, instead of these plots being powered by individual bespoke assays, they're being powered by one single high-dimensional assay. To me, that's the equivalent of going from paper boxes on the kitchen table to a 3D printer. Now, you might be wondering, why are we giving away all this value? Well, there's two ways that we're modulating what we're giving away to protect our commercial interests. One way I already mentioned is the scale of data we're releasing is quite small relative to what we have. But the other way is that we're actually hiding the majority of gene names. So we're holding back and actually making the majority of genes blinded in this data set. And so the use case that we imagine is someone will come into this app with a known compound, they'll search it, they'll see this similarity map, and they'll see at the bottom genes that they are familiar with. It's recapitulating known biology. But then they'll also see these RX-RX3 genes right next to it. That looks interesting. I wonder what that is. Well, in order to find that out, you're going to have to talk to Recursion and get the secret decoder ring. Finally, I want to end on something that Martin actually talked about, and that's the power of these data sets for machine learning. Back in 2009, there's a data set release called ImageNet. And this was about the same time that Martin and the rest of the world, except for a few holdouts in Canada, were ignoring something called neural networks. And those holdouts in Canada, one of which is our scientific advisor, by the way, they said, hey, there's this ImageNet data set. Let's try our approach on that. And they'd been noodling on these things for decades. But once that data set came out, that's when the world started paying attention. They were able to crush the competition with this. And it's really what led to the revolution of deep learning and AI that we've seen in these past decade. Now, these data sets were on the order of 10 gigs to about a terabyte. As we've progressed and done harder and harder things with machine learning, such as autonomous driving, those data sets have grown in size and complexity. There's additional data modalities, and now we're looking at 1 to 5 terabytes. Well, we think with this data set release, we're releasing another revolutionary data set that can power this next level of complexity tackled by machine learning. Again, our data set, we believe, is the largest of this kind, and it clocks in around 100 terabytes. As the machine learning community iterates on these data sets, everyone benefits. For example, when we released RX-RX1 in 2019, we held a competition. And the winner of that competition, we were able to take what they learned, the new methods they developed, and apply it internally and put it into production. Not just that, but we also hired the winner of the competition as well. And as a reminder, while as revolutionary as these data sets might be for the public, we have a much larger data set that we can apply those same capabilities onto. And it's not just more data, but we have different types of data, different modalities. And so it's, and I would also I want to call back to the idea of active learning and using a data engine to not just produce a stacked data set, but to iterate on that. And we see that's what really sets apart the leaders in machine learning, is the ability to generate data at will. So as a recap, the goals of releasing this data set and application are to offer a glimpse and power into our internal data and tools, to pique the interest of potential partners, to provide the largest data set of its kind to enrich the field and foster a generation of next-generation computational biologists, and with that, discover new methods and bring in those along with the bright talent that created them. So with that, I would invite you to visit rxrx.ai to learn more. And we can now break to lunch and the tour. I'll have Ryan Kelly, our Chief Communications Officer, do the logistics. Before I go, though, I just invite you, as you go through your tour, try to invoke your inner child. Be curious and get excited about the possibilities that all this new technology is enabling. Thank you.
We are so excited that you're here today. And one of the reasons why we decided to have it here in Salt Lake, and not a ballroom somewhere, is that there's amazing things that are going on here every day. And we wanted to make sure that you were not only able to hear us talk about these things, but actually see it in action. Thank you so much. Enjoy the tour.
All right. Well, welcome back to the afternoon of Download Day, and thank you all for coming here to spend the day with us. My name's Heather Kirkby. I'm Recursion's Chief People Officer, and I wanted to start by sharing that actually, come this spring, I'll have been with the company for 4 years, and my family still loves to tease me that coming to Recursion was the very end of a Bay Area only job search that I had going on. And the thing was, though, I was 20 years into a career in Silicon Valley. And I knew a couple things by then. I knew what great companies looked like. I had a sense of what great leaders looked like. I had actually started my career in Arctic Alaska, of all things. I was a mechanical engineer collecting geophysical data, so I knew a thing or two about data sets. And so in my Bay Area job search, when I found a company in Salt Lake that was exceptionally special from a company perspective, from a leadership perspective, from a platform perspective, I knew the answer for me was that I wanted to join, that I wanted to be part of building this company. And it's interesting because I can remember those feelings, and sharing Recursion with you today and knowing what your experience might have been like walking through those doors for the first time. I remember my feelings. And the feeling I had this morning hearing people telling our story was one of awe, and I'm feeling that right now, a lot of awe and gratitude. And what I want to talk about for a minute before I kick things off and hand it over to the next speaker, I want to talk about this company's ambition. This company's ambition, and hopefully you've felt it and had a taste of it so far today. It's at the center of everything we do. We imagine the unimaginable. A day like today, 5 years ago, unimaginable, right? Like the biggest transformational biotech partnership in the industry, unimaginable. And yet we do these things. And so how do you cultivate a company, a culture of ambition that continues to deliver on the unimaginable? And I'll give you 2 essential ingredients, and one is the mission, and we keep coming back to it. Like, I expect all of you to be able to walk out of the building today and say, my gosh, Recursion exists to decode biology to radically improve lives, right? And we keep talking about it because it's our North Star, because we're not here to discover a few medicines. We're here to fundamentally change the paradigm of drug discovery and do things at an unprecedented scale. And we have to teach people when they walk in the door that what made them successful in other contexts may or may not make them successful here, and we have to get them to question some of those things, and we have to teach them what makes them successful in our context. And if I hearken back to Lina's first slide, which she said so beautifully, this might be the most important slide, and I would argue yes. It was a beautiful representation of our founding principles. From platform to data to connected data to virtuous cycles and industrialization, those are central to how we approach science and technology here, to decode biology. And the second ingredient that I'll name, and a lot of them are hanging over these super stairs here, it's our people, right? It's the diversity of our people. It's a superpower. I talk about the necessary functional diversity that's required to decode biology. We have as many technologists as we have scientists, and they're accelerated by an incredible group of G&A leaders. It's necessary but insufficient. We have to have a group of people that will challenge orthodoxy and tradition and convention and how things have been done before and in other places. And to do that, we intentionally seek and welcome diversity along the lines of gender and race and ethnicity and sexual orientation and so much more. Because in those different lived experiences are different perspectives and different approaches to problem solving and different ideas to solutions. And in the friction that comes from those differences, good friction, we try to create good friction, comes breakthrough thinking. And that's the path to decoding. Our ambition is born from our mission and our people. And so with that, I'm going to introduce our first speaker, Dean Lee. Dean is one of our very special people. We think of people expansively. Investors are our people. Partners are our people. The board is our people. Dean is the president of Merck Research Labs. Dean's a co-founder of Recursion. Dean is on the board of directors. Dean, come on up. Thank you.
All right, so first of all, I was going to say thanks to all the Recursionauts, but I promised Chris I wouldn't embarrass him too much because I actually know him quite a long time. But we can all do that in private. But it's an absolute pleasure for me to be here, and it fills me with pride and memories. And for some of you who know, I've spent 25 years of my professional and personal life in Utah, so it's great to be here. But I would ask all of you and all of the investors to ask, I would like to remind people that this is in Utah, and it's in Utah for a reason, and I wanted to step back through that reason. But before I do that, I should just remind everyone that Chris, I think you were doing MD PhD at University of Texas, San Antonio, right? So he came here in 2009, and what he's done is incredible. He came here to complete his MD PhD, which he never completed, which as one of the people who run the MD PhD program, there's winner, 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 didn't complete. But with that, I'll just go back to understanding, and I think it's really important that all of us understand what recursion is in relationship to what Utah is, and actually what brought me to Utah, and how when I met Chris, it was like a rebirth of the same idea, but with speed, with rigor that I didn't think was possible. And so I want to do that by sort of explaining the way that sort of Marty explained his own personal journey. So I'm going to take you back a few decades. So I did my MD PhD, which I completed, at WashU in St. Louis. I did my internship, residency, and fellowship there. And I need to point out someone here that some of you Recursion people don't know, and some of your investors. Joe Miletage, can you raise your hand? So Joe Miletage is a major consultant for Recursion, but I'll just tell you how lives are linked so much. When I was an MD PhD program, and some of you will have to ask at some point, how do I know Chris? Well, really the way I know Joe Miletage is not through Joe Miletage. It's actually through his wife, because his wife used one tissue culture hood and I would use the other one. But Joe Miletage was a professor. He had MD PhD at WashU, and he was coming back. He was coming back from San Francisco and starting his lab. And the reason I want to just highlight him is he was head of discovery at Amgen, head of discovery at Merck, and he's the one who essentially trained me. So if things go, for those of you who are investors, if things go sideways at Merck because they have a new president of MRL, it isn't my fault. It's Joe's fault. But I just wanted to sort of emphasize that I remember getting ready to leave WashU, and Joe Miletage will understand this. So WashU is in St. Louis, and it's a great medical school, but they have a little chip in the shoulder. And when you go into where the lecture halls is, there's pictures of every Nobel Prize winner, and you will get pimped as to what each Nobel Prize winner did. That's the culture of that. So I was sitting there going, I'm going to leave WashU, and I'm going to go to Utah. And Joe will understand that, but there are different medical centers and different things. Duke is a yes sir, no sir institution. Harvard's not. Stanford is definitely not. But WashU's a little bit of yes sir, no sir. And so getting called in by the chairman of medicine, David Kipnis, or the chief of cardiology like Bert Sobel, because their concept of if you leave WashU, you have to go to Boston and San Francisco. In fact, Joe went to San Francisco, and I said I wanted to go to Utah. And they're like, now that I speak Utah dialect, it's an oh my heck moment for them, but that's not the lingo they use in St. Louis. But the rationale I did do this is because there was a cycle going on. There was a cycle going on in relationship to science that you could see happening. And I'm going to tell you what I saw there, and then I'm going to bring it back to recursion. You had big data back then. You had a phenomics database. It was curated, collated, and it was connected. And it was only in Utah. It was only in Utah. That was the Utah population database. That was the data. You had algorithms to turn that data into insight. What were those algorithms? There is a very important paper that any of you who trained at the University of Utah best know this paper, because if you ever did rounds with me, I would ask you what are the most important papers that came in Utah. And there was a paper that was by Botstein, Davis, Skolnick, and White. It gave the rationale as to how you would be able to take phenomics data in a population database that was linked by families and how you would find genes associated with disease. And I would remind everyone, and unfortunately Marty's not here, there's 2 Stanford people on that paper, and oh, okay, where is he? All right, there are 2 Stanford people on that paper, and there are 2 Utah people. Let me remind you who actually found genes using that. You have Skolnick and you have White using that to get you BRCA, NF1, APC, long QT, pulmonary arterial hypertension. I'm looking at Jeff right there. That's what you had. You had big data with an algorithm giving you insights that could be used as information. But that information is simply information, because an association is simply an association. If you do not have the power to do an experiment to prove causation, you don't have anything. When Joe and I, on behalf of Merck, go and look at companies and you tell me you have all this algorithm, we're like, yeah, but prove it, show me. And so as you walk around, what I would tell you is the reason I came is that they had the Utah database, they had the ability to do algorithms to get genes that then triggered a hypothesis, and they had Mario Capecchi doing knockout mice to be able to turn that idea, that information, that hypothesis into causation. And only when you have causation do you have any semblance of knowledge. Data, information, and knowledge. And if you do that in a cyclical way, and you can make it a flywheel, just maybe you have a chance at wisdom. So I'm sitting there, and there's a scary office that the chairman of medicine, WashU, can make your career, but can also do the opposite. Is that a fair thing, Joe? I mean, right? And so you laid that out. That's what I came to Utah for. And that played out for how many years, right? From essentially the 1990s to even now. So you sit there and you go, what is it? So they had big data, Utah, they had an algorithm, they could change causation and this. And then I have someone who absolutely knows no biology, has less laboratory skills than Marty does, come and want to join my lab in 2009. But he has an engineering background and he thinks very differently. And when you find out his family and how their family thinks about parallel processing and all of this and how they think about technology, you understand, this guy thinks different than I think. And so again, he comes to my lab, and in some sense, it's the same sort of thing. And so it's this, when you think about what recursion is, it's how do you take big data, in this case, phenotypic data, using the advances of computation and imaging. I would remind everyone the roots of computation and imaging. Some of you may know, but the founder of Pixar comes from, all right? The founder of Adobe comes from, all right? And there are buildings in this. And so that's the sort of view that we had. So essentially what Chris proposed was, instead of doing it human genetics can we do it in systems that could rapidly advance? But it isn't just big data with AI in this. If you do not actually do a causation experiment, and if you do not turn into a flywheel such that you can rapidly iterate three things: iterate your data, iterate your algorithm, and iterate how you do your experiments, you're not going to maximize that. So that's what's happening, right? And the concept is, could we increase the speed but not be sloppy? Could we be rigorous but not rigid? And could we make it a flywheel? So when you listen to what has happened, that's why it fills me with enormous pride to come back to Utah to see what all of you have built. And I'm especially thankful for all of you who have given your time and energy to make this possible. And those of you who have invested, those of you who have risked important capital, we thank you very much. And so with that, I wanted to make sure that we understood what is special about Recursion, its roots within Utah, but I do want to turn it over to the next speakers, because the reason why we're trying to do it is we're trying to affect patients and their lives. Thank you.
Thanks, Dean. Yeah, Utah. It's a very special place. I have the pleasure of bringing up Virani. Shaf is our Chief Corporate Officer, Corporate Development Officer. He's also our Interim Chief Medical Officer. We're grateful for Shaf and how he's doing both of those things extraordinarily well. So he's going to walk you through clinical. And then we are going to take a break after Shaf speaks. So just wanted to let you know that.
Thanks very much, Heather. I think first and foremost, just a big welcome to all of you. Thank you so much for coming to visit us. Hopefully what you heard this morning in terms of some of the advances we're making on the sort of earlier equation as far as the company's technology is concerned, really my job as part of this session is to really bring it home to the patient, right? So just a bit of background. I joined Recursion coming close to about 3 years ago. And, you know, from a background perspective, I've been in the industry for quite a while now, you know. So before I joined Recursion, I was at BridgeBio as a CEO of 2 of the subsidiaries, had good exposure to oncology and to rare diseases there. And then prior to that, a long stint in the Roche Genentech world across the globe, where, you know, we underwent a labor of love trying to create these franchises or medicines outside the field of cancer, primarily in neuroscience, ophthalmology, rare diseases. So it's a true pleasure when I stand here today and we talk about our amazing technology, our amazing maps, the insights that these are generating, which are amazing in their own right, but ultimately we have to run that ultimate experiment, which is, do these insights take us to the patient in a way where these medicines will make a clinically meaningful effect on the patient's journey? We talk about the concepts of industrialization. That's important in several respects. It's important to think about industrialization in terms of scale, and that's because the unmet need across a huge variety of diseases remains acutely strong. There are thousands of diseases today where patients do not have adequate solutions. So scale is important. Industrialization as it relates to speed. I'm sure as you walked into Recursion today, you saw some pictures. You saw some pictures of patients. And every year, we have to take some of those down for obvious reasons. So there is a sense of urgency with which we need to get our insights validated, translated, and converted into medicines to help these patients to move further. And then also another axis of industrialization as it relates to precision. You know, we talked about this morning, we went through, you know, presentations from Lina, from Imran, from Laura. As we're building, as we, you know, scaling up our ability to connect the dots in terms of the phenomics data, in terms of transcriptomics, industrializing transcriptomics, industrializing hopefully at some point proteomics, right? And get closer and closer to what will really move the needle in clinical care of a patient in a responsible way. And when I talk about that, is both the axis of efficacy, but also the axis of safety as well. So one hopes that the ultimate output from all the work we're doing here will give us more effective medicines that can really move that clinically important needle in patients. So before I launch into the clinical programs and give you, some sense as to why we're excited about what we have in mid and late stage development, I just wanted to spend a little bit of time just giving you a preview on how we think about value creation through our pipeline at Recursion. The way, having spent a lot of time in the industry, I think of our pipeline as one pipeline. The pipeline that Recursion executes with our Recursion nodes, and then the pipeline we execute in partnership. Obviously, our mission is to decode biology to radically improve lives. And by definition, that means the ambition is huge across all the verticals of biology. But at the same time, we also have values that push us to getting medicines quicker to patients and to more patients. So we have to be pretty disciplined and pragmatic in terms of how we think about value creation internally and where we join forces with partners, especially in areas of intractable disease, intractable biology, such as neuroscience, fibrosis, where we have consummated some of the largest partnerships in industry, not even moving beyond the sort of sphere of tech-enabled drug discovery. But what we, our collaborations with Roche Genentech, my alma mater, our collaborations with Bayer, this is a true partnership, not just for capital reasons, but bringing in the intellectual horsepower from both organizations to really try and move the needle as far as understanding the pathophysiology of these super complex diseases. So our focus in what we're very intentionally structured is to keep areas like oncology, precision oncology, rare diseases, wholly internal at Recursion. And as I represent the clinical development team today, we're very intentionally building capabilities and infrastructure to really welcome these great early stage programs that Laura just shared with you, Project Alpha, Project Gamma. We're building early stage oncology development capabilities, as well as a formidable translational medicine capability to really continue to hone in the axis of precision medicine and offering solutions to patients that move that clinical needle. So here's a snapshot of our pipeline. And the first thing I personally like when I see this pipeline is that I don't see a fourth-in-class anti-PD-1. I don't see a third-in-class JAK inhibitor. I don't see a 10th-in-class BACE inhibitor, right? And you can see the nature of our pipeline, which, if you actually take a step back and think about what we've heard this morning, this is the essence of our platform, to generate novel insights in biology. And if you fast forward that concept into how does this translate into medicine, it'll typically translate into first-in-disease or first-in-class approaches, right? So this is the nature of the clinical programs. It has made us, for example, partner very, very closely with the FDA, with the regulators, with the patient organizations to really make sure that we truly understand the patient disease journey here so that we can design ultimately, with the great clinical development team we have here, the right experiment in patients to tease out effectiveness. So we walked hand in hand here. 2 of the programs have been granted fast track designation by the FDA, and this will enable us to have more seamless interaction and share data and make data-informed decisions as we move forward. At the same time, these programs represent a huge unmet medical need across the board where these patients, as we speak today, have absolutely zero medical solutions to help them. So let's launch into the programs. The first program, as you heard Dean mention as part of his work, and I'll connect the link there, and we can learn a little bit more about Chris's PhD thesis. But the first program, it gives me great pleasure to introduce our program, REC 994, in the treatment of cerebral cavernous malformations. Now, one thing I didn't mention as part of my introduction is that prior to joining industry, I spent time as a clinical neurosurgeon. I trained as a neurosurgeon. And when I think about diseases like cerebral cavernous malformation, NF2-driven meningiomas, I had a very different solution to offer patients for these diseases, right? It was a pretty crass solution in terms of either resecting the lesions or trying to ablate them using radiotherapy, which has got more precise as the field has moved on. But at the same time, I think the unmet medical need here, when you think about brain disorders, one of the basic things I learned as a neurosurgeon is that the brain is a pretty unforgiving organ in the human body. Anybody who's not welcome to the party, so you have parenchyma in the brain, you have cerebrospinal fluid in the brain, and then you have blood vessels in the brain, and that's it. Nobody else is invited to the party. So when you get something like a cavernous malformation, a brain tumor, an aneurysm, there's always trouble that gets created, right? Trouble in terms of vascular leakage, increased intracranial pressure, focal neurological deficits, inflammation that leads to seizures, headaches, behavioral changes. All these things come and become part of the patient's journey, which tends to be a miserable one. So here we have our first small molecule program that's a superoxide scavenger that's currently enrolling in a phase 2 trial. Here we've been granted orphan drug status both in the US and ex-US. And really sort of delving a bit more into the disease, the unmet need here remains huge. This is the first industry-sponsored program that's attempting to tackle these trouble-causing malformations. We've had investigator-led studies here. We have a study that's ongoing at University of Chicago that's looking at atorvastatin. And then we had a negative study that just read out in Europe, in Italy, using propranolol. So again, more disappointing news for the patients. Agents that perhaps showed some mechanistic promise haven't really moved that needle clinically that we care so much about. What happens with these lesions? These lesions are essentially mulberry-shaped malformations that have very thin walls, and really the problem is twofold. One, they have a finite risk of rupture that can lead to intracerebral bleeds, that can lead to high morbidity, quite often to mortality. The other integral part of this disease, which has been poorly understood by investigators, by clinicians, and also by regulators, is what happens when there's vascular leakage, when you start finding blood deposits around the actual lesions, and this starts a cascade of inflammation, and what sort of signs and symptoms does that lead to? So when I first came across this program and had to have regulatory interactions, the first interaction was with the CV Met division of the FDA, and they wanted to talk about stroke. Now the stroke risk here is 0.4 to 0.6% on an annualized basis. And what we know about cerebral cavernous malformations is that it gives you a finite stroke risk, but that's certainly a small part of the patient's journey. And the CV Met division wanted to talk to us about stroke scales, MRS, and the National Institute Stroke Scale, which to us was not really fully appreciating what the patients were going through. So strategically, we eventually managed to pivot this program into the neurological division of the FDA, where again, arm hand in hand with the Angioma Alliance, with the Patient Foundation, with some patients actually taking the regulators through the journey. We've had very robust discussions to date on what exploratory endpoints we could look at here. At the same time, both patient-reported outcomes as well as clinician-measured outcomes as well. So we're paving the ground here. Nobody's developed a drug. There's no regulatory precedent. There's no precedent in development for cerebral cavernous malformations. Is this a rare disease? I mean, you can look at the numbers there, right? I mean, the way we think about rare diseases, often we think about smaller populations. We've seen amazing progress and fantastic for patients to see advances in cystic fibrosis, more recently in spinal muscular atrophy area I was deeply involved in at Roche with the PTC Therapeutics deal there. But when we look at CCM, I mean, the numbers are impressive here, right? We have up to like 360,000 symptomatic patients today in the US and 5 key European markets. As MRI gets more frequently adopted into the evaluation of neurological disease, becomes more accessible, becomes cheaper, these numbers will only go up, right? Both in terms of incidental findings of CCMs, at the same time, the propensity of these lesions to eventually start causing clinical symptomatology. Today, given those caveats with regards to readily accessible imaging, it's approximately over 1 million patients today worldwide live with these lesions today. There's two forms of the disease. There's a familial form that's caused by aberrations of the genes CCM1, 2, or 3, and this represents roughly between 20 and 30% of the whole CCM pool. At the same time, as I mentioned, this is inherited in an autosomal dominant way, and when we compare it to the other diseases, the numbers here are exponentially large. Again, no approved therapy as it sits today. It's primarily supportive care. Patients have headaches, they have medicines for headaches. Patients have seizures, they have anticonvulsants to help control that. And the seizure frequency can vary. There's patients who have seizures on a weekly basis, on a daily basis. So it's a real crippling disease in terms of quality of life, ability to drive, and the things that we normally take for granted. So this is a good point for me to now delve into the science of this disease via introducing no one other than Chris Gibson to give you a little preview into his work that led to the formation of Recursion.
Thanks, Shaf. Thank you so much. I'll go through a quick summary of my dissertation, but before I do, I want to just talk about something really important here. Why is nobody else going after this disease? So the genes that encode this disease, or cause this disease, encode scaffolding proteins, which are not considered druggable by most folks. And in fact, I was at a conference a couple of weeks ago with one of the heads of research of one of the top 10 biopharma companies. And I was talking about this program, and he said, Chris, I love what you guys are doing at Recursion, but the reason none of us are going after this is because we know that no medicine is going to revert these lesions, these aneurysm-like lesions in the capillaries of the brain, back to the normal capillary. And I said to him, we don't need to do that. We don't think we need to do that. We think we need to slow the growth of these lesions. We need to reduce the number of these lesions over time, but most importantly, these are not high-pressure lesions. These are not lesions that are growing in the arteries or even the larger veins of the brain. These are growing in the capillaries. So the law of Laplace here does not apply so much as if these were real aneurysms growing in high-pressure vessels. In fact, what we need to do is actually reduce the activated sort of inflammatory milieu that surrounds these lesions. In fact, there's some fantastic papers that show in these patients, their symptoms arise most typically when they have some other comorbidity. So a patient might go for 6 months without seizures, and then all of a sudden they get the flu, and all of a sudden their seizures are hitting off because these lesions are inflamed, they get leaky, you get hemosiderin deposition, which leads to this feed-forward inflammatory milieu. And so we believe at Recursion, if we could start to reduce that activated state of the endothelium, one could actually go after this extraordinary opportunity to bring a first-in-disease treatment to one of the largest areas of unmet need remaining today in the rare disease space. So how do we get there, especially given these are undruggable targets? And unfortunately, I think probably like Rob called Dean, so he had to hop on a call real quick. I was going to make a lot of jokes about Dean and how bad he was in the wet lab. But what Dean was able to do in his work at the university was partner with a small number of other labs to really create a state-of-the-art of this field. And in 2011, this is what it looked like. This is from a Nature Neuroscience review. This is what people had figured out about CCM. And Dean's lab was one of the labs that helped uncover, one of the reasons he came to Utah, that CCM1, CCM2, and CCM3 are the genes that drive this disease. And in his laboratory, we had actually helped to hone this hypothesis using traditional molecular and cellular biology tools, that the disease was being driven by activation of this protein called Rho-A. And so what we actually did was take a very, very simple approach to mitigate the activation of Rho-A using a really common drug called simvastatin, which through HMG-CoA reductase will mitigate the activation of Rho-A. And we put it into these incredible animal models that Dean's lab had built that are almost like mini CCM lesions just in a mouse, which is not always what happens in animal models. And we were so excited to read out this data. And I remember being in lab when Aubrey Chan, the student who was leading this program, read out this data. So after about a year of doing this experiment, we had statistically no effect on the lesions. There was a strong trend here, almost a statistically significant trend towards increasing the number of lesions in these mice. That was a devastating moment for us. We felt like we were on the cusp of making a real breakthrough for these patients with a medicine that was going to be extraordinarily inexpensive and hopefully have a benefit to them. But it caused us to reevaluate our process. And when we were doing that work, we noticed something. We noticed that when we modeled this disease in human cells, human endothelial cells, like the ones that are probably running through our pipeline right now at Recursion, the cells looked really different when you modeled the disease. You can see the difference yourself. You don't have to be a pathologist, but you can see that these cells look different. And so we wondered whether we could take an unbiased approach, whether we could get away from this traditional molecular and cellular biology approach and ask, is there some drug, regardless of its mechanism, that can make these 6 cells, SICCM2, look like the healthy ones, SI control here? And so we got to work. And because I was so bad in the laboratory, as Dean made sure to mention, I tried to use as many robots and software as I could to run this project. And so what we actually did was found some incredible open source software from a woman named Ann Carpenter at the Broad. She really pioneered this field. We got very small scale robotics, which was my hand still, but a 16 channel pipette, which was better than a 1 channel pipette at the time. And we started screening about 2,000 FDA approved bioactive molecules or molecules that were commercially available that we thought could give us some insight into this disease. And let me show you what we found. So here's the same images, SI control, SICCM2. We want to make the green one look more like the less green one. For those of you who are purists in the field, you'll know Rho activation causes those green actin stress fibers. So, we knew that was happening. And look at simvastatin. Simvastatin was flagged by this cell profiler algorithm as a rescue of this disease. And you can actually see, if you look in the middle of those cells, there's less of those green tiger stripes. And this is actually, consistent with the hypothesis, but we'd already done the animal model experiment. We already knew that it wasn't working. Our algorithms also pulled out 2 other drugs. Cholecalciferol, I don't know if anybody's aware of this. This is vitamin D, which is like the only vitamin that survived all the trials showing it actually has some utility in folks at higher than normal levels, and REC-994. Now, what's most interesting to me is that to my eye, neither cholecalciferol nor REC-994 look to me like they're rescuing. And this was the scary part of my dissertation, because I had spent a lot of Dean's research money on this project, and the computer was giving us back hits that looked like they were not going to work. But we pursued, and we decided to finish the experiment and ask the question, was the computer seeing something that we had missed? And it turns out that it was. Because as we took both vitamin D and REC-994 into these animal models, just a few months after the failed experiment with simvastatin, both of those molecules delivered a reduction in the number of lesions, both in mice driven by CCM1 mutations, but also in mice driven by CCM2 mutations. Together, those making up about 80% of the causative mutations of patients in this field. And what's more, when we did other preclinical work looking at this hypothesis of vascular stability, we found that there was a deviation in acetylcholine-induced vasodilation, which you can see here on the bottom left, which was completely rescued with REC-994. We saw that there was a dermal vascular leak in the animals that you can see on the bottom right, consistent with what we know is happening in the brain with these vessels being leaky, which we could almost completely rescue with REC-994. And it was these data that gave us the confidence not only to pursue this program, but actually for me to finish my dissertation to leave medical school to start Recursion. And what's funny about Recursion is we didn't work on this particular program for 3 years after that. We didn't have the resources to go try and explore that first hypothesis I talked about this morning, and to try and build a translational and preclinical team that could take the medicine to the clinic. But the founder of NPS Pharmaceuticals, who also is based here in Salt Lake, told me one day, he kind of put his arm around my shoulder and he said, Chris, it's going to be important that you actually take medicines to the clinic at some point. You should probably go back to that compound and advance it. And so we licensed it from the university, and Recursion was really starting to get rolling on our platform. And so we were then in a position to advance this program forward. And so I won't go through all of the detail here, but suffice it to say, for those who understand endothelial biology, reactive oxygen species are a very well-proven activator of the endothelium. If you have a lot of reactive oxygen species in the endothelium, the cell-cell junctions break down, the cells become inflamed, and you get all kinds of things which are very consistent with what we see in the context of this disease. What this drug does is scavenges superoxide, which is the predominant reactive oxygen species that arises when you lose CCM1 and CCM2, which we were able to determine is driven by changes in both superoxide dismutase 2 and FOXO1 transcription factor levels.
And we believe that if you can mitigate these effects, you can dramatically reduce the inflammation in these cells, leading to a more stabilized quiescent endothelium, which in the blood-brain barrier is so critical to help reduce that feed-forward inflammatory milieu. So I'm very excited to turn it back over to Shaf to talk about the phase 1 trial results.
Thanks, Chris. So just bringing it back to development. So you heard a little bit about the mechanism of action, what we think this particular drug is doing. And of course, our first step has to be to put it into a complex human system, in this case, healthy volunteers, and see how the drug behaves. As you'll hear as I go through the clinical programs, quite often when we put known or NCEs into the first healthy volunteer study, it's a moment of trepidation, right? Because these drugs have never been into a complex human system. So everything from how it's going to behave in terms of pharmacokinetics, in terms of exposure, in terms of tolerability, safety, all become areas of high vigilance for us. Here, I'm very glad to report this drug has got a very benign profile. We dosed all the way up to 800 milligrams, both in the single and multiple ascending contexts. And you can see the numbers there, right? In terms of serious adverse events, absolutely nothing seen there, no drug discontinuations. So first hurdle, drug has got linear pharmacokinetics, great exposure. Certainly when we relate it to the levels we need to try and modulate the disease, we've got a large margin here to work with. Let's get to the clinical trial. So this is a super exciting clinical trial from my perspective. As I mentioned, this disease has never had anything in development, anything thought through with regulators in terms of what does the path forward look like here. Two things I want to point out about this trial. It's an exploratory phase 2 study that's really looking at a couple of things. It's looking at what has been measured before, things like the MRS, things like the National Institute of Health stroke scale. We know those entities, we know how they behave in disease states that are not quite CCM. At the same time, we've also done a lot of work in the background, really trying to understand the patient journey further. So here come in the patient-reported outcomes. Let's talk to the patients. Let's find out what they're going through. Let's find out what they're suffering from, okay? Headaches, seizures, focal deficits, double vision, behavioral symptoms, anxiety, stress on that, right? So let's try and get composite scales really validated. And the work we've done here has been with the University of Rochester to really come up with a pretty comprehensive 19-domain, 100-questionnaire patient-reported outcome, which we'll study as part of this disease, or part of this phase 2 trial. For neurological diseases, again, thinking about surrogate endpoints, here, this is not just standard MRI imaging. Chris mentioned what happens pathophysiologically in this disease. You get this leakage, vascular leakage that leads to hemosiderin deposition. I'm glad to say that a consortia of investigators have done a lot of work validating a special protocol of MRI called the QSM, which is quantitative susceptible mapping, which is as powerful as an in vivo, a real-life in vivo study that really tracks from a temporal perspective, a time perspective, exactly what's the axis of this hemosiderin deposition and how does this affect any perilesional edema and inflammation. So we're measuring that in a subsection of patients as well. So we hope to get some sensitivity with the imaging modality there as well. We plan to enroll 60 patients, 2 dose groups, 400 milligrams placebo-controlled, and we're going to treat for 12 months, after which eligible patients will enter into a long-term extension study. Enrollment is progressing well, I'm glad to report, and we expect top-line data in the second half of next year for this phase 2 program. Moving on to the next clinical program, this is REC 2282 for the treatment of progressive NF2-driven meningiomas. This is an HDAC inhibitor that's a small molecule, has got some very special properties, and I'll talk through those as we compare this drug to the landscape of HDAC inhibitors. This is a unique trial. It's an adaptive phase 2/3 study, so it gives us a unique opportunity to be able to get, hopefully, an effective drug quickly to patients. Another highly underserved disease, brain tumors, malignant or benign. there are very few options out there. When we look at the brain tumors caused by mutations of the NF2 gene, meningiomas, vestibular schwannomas, or also known as acoustic neuromas, ependymomas, both occur in the brain and in the spinal cord. As we speak today, no therapeutic agents have been approved. There have been, unlike CCM, industry has tried to test different modalities, different anti-cancer agents to see whether we can move the needle clinically in these patients. So far, nothing has really met the mark. The disease itself is an autosomal dominant tumor syndrome resulting from an inactivation of the NF2 gene. And really, it's again like CCM, it's inherited either, it's autosomal dominant condition, and you have familial forms or sporadic forms as well of the disease. As I mentioned, there's different manifestations in terms of brain tumors, their locations, and this, the way I think about it is, the actual disease itself, due to the loss of NF2 gene, gives us a real life cycle expansion opportunity. Our first forays into NF2-driven meningiomas, but mechanistically, and as far as the mechanism of action of the drug is concerned, we can certainly look at indication expansion into the other tumors I just mentioned, the schwannomas and the ependymomas. Standard of care is surgery. Now, again, coming back to my sort of neurosurgery days, this is probably the most tricky brain surgery to do for tumors. When one is conducting surgery for something like a glioblastoma grade 4, where the prognosis is dire, typically 6 months, one has more latitude in terms of risk-benefit, in terms of how you treat eloquent tissue, fully functioning brain tissue around the tumor. For meningiomas, these are benign tumors, but yet at the same time, to stop them from coming back, one has to excise up to a 1 centimeter margin of normal brain tissue. Now, I don't know how you guys feel, but if somebody was going to take 1 centimeter of my functioning brain tissue out, I'd be quite concerned, because usually you have collateral damage when that happens. So surgery comes with morbidity, often paralysis, often focal neurological deficits. And what we see in this genetic syndrome of NF2-driven meningiomas, often you get multiple meningiomas that show very heterogeneous behavior in terms of growth rates, right? So you can imagine the prospect of recurrent surgery is quite common here. So the unmet need is commensurately even higher. Presentation typically of an uninvited guest in the brain, as I mentioned before, a tumor in this case, that can lead to focal deficits, can lead to seizures, can lead to raised intracranial pressure that can lead to death in case of multiple large meningiomas. Double vision, loss of visual acuity, certainly in the case of schwannomas where they're located, hearing loss is a prominent presenting sign there. Again, coming back to how many patients suffer from this, we estimate today to have around 66,000 patients who harbor these meningiomas, of which about 33,000 of them will be familial or will be sporadic meningiomas that have a demonstrated NF2 mutation. So the treatment population here is quite large as far as normal rare disease sort of numbers go. Primary intervention, as I mentioned, is surgical excision, and certainly a lot of the times these tumors occur in areas, at least from an anatomical standpoint, are not amenable to surgical excision. Getting skull-based meningiomas is quite common. Sometimes we see meningiomas that impinge onto the brainstem. That tends to happen, in which case focal radiotherapy is the only option to control these tumors. Let's get back to our operating system. How did we come across this? So in the early days, when we look at knocking out a gene, in this case NF2, and we look at our Prometheus plots that look at, okay, you can see in red here, these cells represent HUVEC cells where NF2 has been knocked down. And then we do mass phenotypic screening to see which agents actually revert these disease cells back to the green ones, the normal, healthy, functional state. And here we found REC-2282, a concentration of around 1 micromolar, very consistently brought these disease cells back to a healthy phenotype. So this gave us the first reason to believe that we have an agent here that could actually show promise in the treatment of NF2-mediated tumors. Mechanistically, loss of NF2 leads to loss of a key function called Merlin, a key protein called Merlin. Merlin is absolutely critical in tumor signaling. the pathways that are involved are the PI3 kinase, the AKT, and the mTOR pathways. And really what happens with aberrant mTOR, PI3K, AKT signaling is you start getting tumor growth. And HDAC inhibitors essentially block the protein-protein interaction with AKT and lead to suppression of this. So mechanistically, it makes sense. The second reason to believe. We then came across and learned through an open-label phase 1 study that looked at both CNS tumors and peripheral solid tumors and looked at the effect of this particular HDAC inhibitor. And I'll get to that data, but before that, we did additional profiling and we looked at the effect of this drug in traditional in vivo setting. So we looked at vestibular schwannoma xenografts in nude mice, and you can see that on top, you can see the, when we give vehicle, you don't really see an effect there, and then a very consistent effect in the growth of these xenografts and the suppression of growth of these xenografts when you give Rec 2282. At the same time, if you look at a bioluminescence-led readout here in meningioma cells, you can see pretty marked suppression of the signal, and then of course upon stopping treatment, we see some durability of effect, durability of response there. And then we get to this phase 1 study I just mentioned. Here again, this study was not designed to provide statistical power of any sort. It was a signal-seeking study, but really encouraging to see two things here. One, we see that the NF2 driven central nervous system tumors, we saw a progression-free survival here of around 9 months compared to around 2 months for non-CNS tumors. And really what was encouraging to see for an HDAC inhibitor was that there were patients who were tolerating this agent for a duration of up to 27 months. So this gives us the second tag, the second hook to say, okay, we may have an agent here if we thread it carefully through a deserving patient population here, at least from a clinical disease standpoint, that we could have something that will give impact. A little bit more about this compound, Rec 2282. When we compare it to, I think there's about 5 approved HDAC inhibitors in the market today, all have different characteristics. There are HDAC inhibitors that are given intravenously. There are HDAC inhibitors that are orally bioavailable. What we see in 2282 perfectly fits the ideal target product profile for what we would want for a CNS tumor. Orally bioavailable, targets the pathway effectively, orally administered, and has CNS exposure, and then hopefully no QT cardiac liability, which we consistently see with the other HDAC inhibitors. Let's get to the trial here. Again, this is the first in-class HDAC inhibitor for the treatment of NF2-driven meningiomas. What we require the patients to have here is MRI confirmation of the tumors. We're targeting both the sporadic form, in which case the patients need to have documented diagnosis of an NF2 mutation through tissue biopsy that could have been done through previous surgery, or a confirmed diagnosis of NF2 in familial cases. Outcome measures here are pretty standard for brain tumor studies. We're looking at progression-free survival at the 6-month mark. We're looking at tumor volumetrics as far as overall response rate, which will constitute of partial response and complete response, and we'll be measuring these tumors. What's interesting thing about this trial is it's targeting progressive meningiomas. So the patients will have to show evidence of a brain tumor that is showing progressive expansion. And the way the criteria have been set so far is they have to show 20% annualized tumor growth before they can get enrolled in the trial. Initial open label phase to enroll 20 patients, 2 doses, 40 and 60 milligrams, and then we'll undertake interim analysis and further FDA consultation on the confirmatory phase 3 design of the study. Enrollment is progressing well here as well, and we expect the interim analysis primarily targeting safety in 2024, next year. Getting away from the brain, now let's move on to FAP, our third clinical program. Here, this is an interesting story because, we've got now 2 indications that I'll talk about. Familial adenomatous polyposis, and then solid tumors, very recently harboring exon 1 and APC mutations. There's a lot of commonality as far as the pathway biology is concerned, which I'll touch upon. Here we have an inhibitor of MEK1/2, an oral small molecule inhibitor. And again, this program, with extensive consultation with the oncology division of the FDA, has garnered fast track and orphan drug designation status both in the US and ex-US. Another genetic disease or predisposition syndrome that leads to significant morbidity, often to mortality. Here we have an autosomal dominant tumor predisposition syndrome, goes by a mutation in the APC gene. It's a classical germline mutation that results in thousands, hundreds of thousands of polyps emerging in the GI tract. Typically, they start off when folks in the adolescence, the juvenile form that leads to hundreds of thousands of colonic polyps. So you can imagine, just not in addition to the risk of these polyps progressing to frank colorectal cancer, which is 100% if left untreated. At the same time, you can imagine that amount of disruption in terms of colonic function, what functional symptoms these patients have in terms of gastrointestinal transit syndrome, in terms of diarrhea, in terms of just general sequelae of inflammatory disease in the colon. As I mentioned, 100% progression to frank colorectal cancer if left untreated. We estimate today approximately 50,000 diagnosed patients in the US and in the 5 key European markets. No drugs approved to date. There was a fair amount of work done on celecoxib a while back, and certainly that drug was approved with the caveat that some key post-marketing studies were to be conducted, which never were, and hence the approval was withdrawn. So as we stand today, not a single agent or therapeutic class has been approved for the treatment of FAP. The option patients have is a rapid path to pretty mutilating surgery, which involves initially a pan-proctocolectomy to remove the susceptible lesions and to try and reduce the risk of progression to frank colorectal cancer. Also, these patients go through a pretty intensive schedule of surveillance, right, through endoscopy, sigmoidoscopy, pretty much on a 6-monthly basis to try and monitor the status of the disease. Again, coming back to some familiar pictures you've seen here, again, when we knock out in our system the APC gene here, and we get the disease cells due to the knockdown, and we screen. In this case, it was a pretty extensive battery of MEK inhibitors and ALK inhibitors that was screened here, up to about 1,500, I believe. And we certainly included agents such as selumetinib, binimetinib, and we found, even given that REC 4881 provided the most potent and specific rescue of these cells where the key gene APC was knocked down. So what's going on here from a pathway perspective? And this biology is common to both FAP and the Exin-1 program in solid tumors that I'll introduce next. And really here, the case is one of dysregulation here, right? We've got the beta-catenin in the Wnt pathway, the beta-catenin degradation complex. It's really a surveillance complex to try and stop activation of RAS. Activation of RAS lights up the MAP kinase pathway that eventually leads to the upregulation of MYC that leads to tumorigenesis. We see here that both Exin-1 and APC are key components of the beta-catenin degradation complex. And what happens with loss of function of Exin-1 or APC is that this complex stops doing its surveillance job and lets RAS continue its aberrant signaling and leaves the MAP kinase pathway unchecked to upregulation of MYC that leads to tumor formation. So any intervention that targets either elements of the complex through the beta-catenin or the Wnt pathway or targets the MAP kinase pathway has potential to actually arrest the growth of tumors here. In our case, we're targeting MEK here, a key component of the MAP kinase pathway, and this eventually leads to downregulation of MYC and suppression of tumor growth. Does this translate? Well, let's go to the in vivo studies, and this is probably one of the most impressive in vivo data sets we have here at Recursion. Here we have a validated, recognized mouse model. It's called the APC-Min mouse model, where the APC gene is knocked out. And you can see here, after 8 weeks of treatment, we see a potent reduction in polyp count. We've obviously benchmarked this against with celecoxib, one of the only other known agents that actually impacts this biology. And in a nice dose-responsive way, we actually reduce the polyp count across the board in this relevant genetic knockdown mouse model. What's even more impressive and excites me even more in terms of the potential clinical translation here is when we look at the graph on the right. Here, when we look at the progression of dysplasia, looking at the number of high-grade adenomas, we see with vehicle there's absolutely very little effect. Again, we see very little effect with celecoxib. But when we look at Rec 4881, 3 doses, a nice dose response in the arrest of dysplasia. This is important because the reason these patients have colectomies and mutilating GI surgery is to ultimately reduce the chances of progressing to frank cancer. And so it's very reassuring for us to see we are reducing polyp count, but at the same time also downgrading the actual grade of dysplasia that occurs in these adenomas. What we did after that, which is the first time we're actually sharing this data, is run a dedicated phase 1 study to get a better sense for what the PK/PD is. I'm sure a lot of us here are familiar. Some of the challenges with trials in oncology is that often we don't have the recourse based on the mechanism of action of the target being modulated to go straight into healthy volunteer studies. In this case, it made sense because we needed to get a better sense for exactly what is the therapeutic index here, how is this MEK inhibitor tolerated, and what doses would give us the optimal reduction of pathway biomarkers such as phospho-ERK to then enable us to move rapidly into a phase 2 trial to make sure that we got the dosing right here. So again, here we tested doses out, obviously checked for food effect, which I'm glad to report we did not observe a food effect here. But here, doses from 4 to 12 milligrams were tested in healthy volunteers. The drug was well tolerated. At the same time, we also ensured that we could match the data to what Takeda had generated before we licensed the molecule in. Getting to our trial, we initiated this trial in third quarter of last year. Again, from an enrollment criteria perspective, we're looking at patients who have a confirmed APC mutation, and we're targeting an aggressive disease patient pool here, patients who've already undergone a colectomy. So here the risk is that they will continue to develop polyps either in the rectal pouch or in the upper GI tract, in the duodenum, the ampulla of Vater, etc. And this is an aggressive form of disease because if you start getting polyps appearing post-colectomy in the rectal pouch, in the duodenum, the next line of surgery is pretty debilitating and happens pretty aggressively. So we have more pinpoint measures in terms of how this agent could potentially ameliorate the progression of disease. Part 1 of the trial will be looking at pharmacokinetic metrics, exposure metrics in these patients who've had their colons removed to absolutely make sure that the exposure, the PK, remains consistent with what we've learned in the healthy volunteer setting and to the data that Takeda generated, and then rapidly move on to the randomized phase with 136 patients across 3 doses, placebo control in part 2 of the study. We've made several protocol amendments here to do a few things here. This is a tricky population to recruit. We've had extensive consultation with our KOLs from the Mayo Clinic, from MD Anderson, with the FDA as to how do we make this study more fit for purpose for patients, most importantly, for clinical sites, for investigators to be able to recruit. So we made some recent protocol amendments here, and at the moment, we haven't recruited a single patient here as yet, but we hope to get sites initiated very rapidly. We have the first site initiated, and then we have a slew of academic sites where we think a lot of these patients who've had post-colectomies will present themselves for enrollment to the trial. So looking forward to the next update where we'll have patients who are actively enrolled in the study. We've also changed the protocol after extensive consultation. Initially, this was a 12-month study. We've reduced that to a 6-month period of treatment. So that should certainly ameliorate, some of the few delays we're having in terms of site initiation. And again, we'll provide an update, at a later stage on this program. Just bridging on to the same pathway biology, a program we announced fairly recently. And what's exciting about this program is it's still Rec 4881, our MEK1/2 inhibitor. This time around, I get the pleasure of bringing in inference-based drug discovery, right? So as you heard this morning, how we go from maps to medicines, how we have these whole human genome level maps. We overlay small molecule perturbations on these, and very rapidly we can generate insights via inference and look at the potential utility of a drug for a particular mutation. Here again, the biology is common, dysregulation of beta-catenin, destruction complex. You saw in the previous diagram the critical role of APC and exin-1 in keeping that complex intact. And here, when we look at exin-1 mutated cancers, there's absolutely nothing, there's no solution for these patients out there. These often are highly resistant tumors, and when we look at across the whole spectrum of solid tumors, we go from lung to hepatocellular carcinoma, where exin-1 mutations as drivers are incredibly relevant. At the same time, then we look at endometrial cancer, bladder cancer. Overall, we're looking at a patient population of around 32,000 patients here. Again, happy to report that with all the advances made in genomic screening, we found that exin-1 is well represented today in all the industry panels. So in terms of patient diagnosis, we're working very closely with big data partners here to learn about, both on the data and the sequencing side, which we'll need for this trial, as to where these patients are and how do we operationally bridge to run an effective phase 2 study. As I mentioned, this is a poster child from our pivot to inference. You can see here, I think the first reassuring thing and encouraging thing to see here is that both APC and exin-1 cluster together, right? So that's incredibly comforting to see with phenomics as the distal endpoint here. I think the second point to bring out here, which you see in blue, across doses from 0.1 micromolar to 2.5 micromolar, we see REC-4881 very potently rescuing the loss of function of APC or exin-1. So this gives us reason to believe that we have an intervention here with a well-profiled compound to enter into solid tumor study. As we speak today, we're actively in the design phase of this study, and we'll provide updates once we've homed in on a formal and final study design. Again, here we ran some validating in vivo studies here. You can see the waterfall plot where we look at mutant exin-1 mice compared to wild-type mice. And you can see a pretty reassuring tumor growth inhibition of around 70% in the mutant setting compared to around 50% in the wild-type setting. Certainly when you look at the related Kaplan-Meier survival curves, one can see the actual delta coming out quite nicely at a statistical significance between mutant and wild-type based on knockout of exin-1 or APC. So again, here from an update perspective, finalizing the design for 1B2 trial. We'll update when the trial design has been finalized. At the same time, we're also thinking about this agent in this context of solid tumors, both as a monotherapy, but also in combination with potentially different agents, potentially with checkpoint, with immunotherapy, in combination with chemotherapy, and in combination with other targeted agents. All these considerations are undergoing, rigorous evaluation as we speak. Finally, changing gears, getting away from tumors, moving on to what I'm proud to say, our first de novo Recursion operating system generated NCE, or novel or new chemical entity program, REC-3964, for the treatment of C. diff colitis. Here we have a small molecule, the first in class small molecule toxin inhibitor for Clostridium difficile that currently is in phase 1 healthy volunteer studies. Another serious disease, right? Clostridium difficile, I'm sure all of us are familiar with it. We've learned about it in terms of the socioeconomic impact it has, especially in elderly populations, in immunocompromised populations, in patients who are undergoing surgery, patients who are undergoing broad-spectrum antibiotic therapy. And really what happens is broad-spectrum antibiotic therapy essentially disrupts the microbiome and the gut flora that opportunistically enables Clostridium difficile to come in. What then happens is the toxins are released and the cascade of inflammation, localized colonic inflammation starts and has the potential to get systemic and make patients very unwell, both from a localized gut inflammation to toxic megacolon to systemic inflammation response syndrome that can lead to tractable levels of morbidity and mortality. Around 500,000, 500,000 cases seen for patients suffering from this disease. And really, the key, there's three aspects going here in C. diff development. There are agents that target the bacteria itself. We're all familiar with vancomycin, with metronidazole. More recently, there's been agents that target the homeostasis in the gut through modulation of the microbiota, through FMT, for example. That's still awaiting approval as we speak. Then we've also had large molecule toxin inhibitors that have also been approved and have had niche positioning, within the treatment paradigm for patients. What we feel we have here is a first-in-class small molecule toxin inhibitor, so far very easy to manufacture, very low cost of goods. At the same time, from what we're learning from the phase 1 study, has the potential to have very good tolerability and good druggability, as you'll see from the data that I share. Significant unmet medical need here with a large cost burden and socioeconomic impact. Recurrence of this infection occurs despite of antibiotic therapy. 20 to 30% of the patients recur, and a pretty tangible mortality. Close to 30,000 patients each year die in the US from uncontrolled Clostridium difficile colitis. Approximately 730,000 patients, US, 5 EU. And really here again, what gave us the reason to believe is, in this case, not a gene knockdown, but actually applying the C. diff toxin to both HUVEC cells and gut epithelial cells. And again, you see the familiar disease state cells when you apply this toxin, and then when you apply a series of molecules through phenotypic screening, we see nice reversion back to the healthy phenotype here in green. And you can see the difference between when this is very apparent to the naked eye when C. diff toxin is applied compared to healthy control. What's going on here mechanistically? It's a novel chemical entity, a new mechanism. So what happens here is the bacteria colonizes the gut. It releases toxin that gets endocytosed into the cytosol. What then happens is this autocatalytic cleavage that releases the toxins, glucosyl transferase enzymatic domain, into the cytoplasm of the cell. This then affects a key event in the pathogenesis of C. diff. What happens here is that it converts a set of row GTPases from the active state to the inactive state, and this then leads to key cytoskeletal changes, apoptosis, and dysfunction of the gut barrier that starts the inflammation cascade, and that goes unchecked unless the bacteria is treated, the toxin levels are diminished, or the microbiota is enabled again to try and remove the offending bacteria. What the drug does is it specifically affects the catalytic activation of the toxins glucosyl transferase, but not the host proteins. It's a very specific effect that we feel will have a real chance of actually stopping or impeding the cascade of inflammation that starts locally initially within the gut and then rapidly progresses on to lead to the pretty serious sequelae of colitis, megacolon, and then systematic inflammation as well. And you can see that here in terms of where the drug works in this key step of affecting the row GTPases to convert them, to stop them being converted into inactive state. Again, the key question, does this translate, right? So we get to the models. Again, fortunate here to have a very relevant hamster model for C. diff infection. When we look at barrier integrity, again, in a nice dose-responsive manner, we see the agent restoring barrier integrity, which is key for overspillage of inflammation to not to occur. At the same time, then we look at some real-life functional data in terms of the Kaplan-Meier. You can clearly see the separation quite nicely of when the drug is given, and that extends the survival, that even extends beyond once the drug has been stopped as well. So this is reassuring evidence for us to then put the NCE into a phase 1 study in healthy volunteers, which is shown here. This trial is currently underway in the Netherlands. Both single and multiple ascending parts that have been initiated are well underway as we speak. And I'm pleased to inform a couple of things. One, what we're learning so far in terms of exposure, in terms of PK, in terms of safety, tolerability, we're seeing very encouraging signs so far, both of this NC acting as a drug in a very dose-responsive manner, giving us linear exposure, PK, but at the same time showing very good tolerability up to doses of 1,200 milligrams, right? So those MAD portion is still continuing for that, and we expect, both the safety and PK readout in the second half of this year. I think another interesting thing with the recent JP Morgan is we've had significant amount of incoming interest from some big pharmas on this program. So we'll initiate those discussions, and we'll see whether we prosecute the development of this particular agent ourselves or in partnership. Those will be good conversations to have. So with that, I will close the clinical section. Look forward to any questions during the Q&A session. But Heather, back over to you.
Like all of you, I'm experiencing today and learning a lot myself. It's so amazing to go deep on the progress. One reflection I was having listening to Shaf was the rate of progress that happens at Recursion. It's really hard to convey when you're here with us at a moment in time and a point in time, but I was trying to think of how to express it, and I was imagining, if it was a few years ago and we were having a day like today, clinical programs probably would have been 10 minutes on the agenda or something like that, right? And another anecdote I would share is when I joined the company, I really wanted to explain to my family what we did here. And it took me about 6 months to really nail the story, and then I was able to tell the story, and the company had changed so much and made so much progress in 6 months. I had to continue to learn what we did, and we often talk about, do our people and are we learning and growing as fast as the company is progressing? And it's actually a really sort of real aspect of being here and working here, and working at the pace of Recursion is something we talk about a lot. I'm gonna start saying a few words to introduce our speaker, Matt Kinn. Matt. So we've spent a lot of today getting to know and understand about the capabilities of Recursion's data set, right? How we map and navigate it. You've come to understand our pipeline, our programs. This is a really exciting moment where we're going to transition, and we're going to talk about the power of that data set and the power of mapping and navigating it in combination with our partners. And we care as much about their pipeline as our pipeline. It's really part of the value that we're trying to deliver as a company. And so with that, I'm going to bring up Matt Kinn, Senior Vice President of Business Development and Corporate Initiatives.
Thank you very much, Heather. I'll pick up on a similar slide that Shafique already spoke to. We've been talking in the afternoon about how we extract value from the Recursion operating system. And we do so in two primary ways. As Laura earlier today and Shafique just most recently spoke about, our internal pipeline is a wholly owned pipeline where we're advancing medicines in oncology and rare disease, two areas where we can move quickly and cost effectively from these early biological insights into clinical candidates and quickly into a patient population as well. In addition, we're building pipelines with external partners through transformational collaborations in large, complex, and resource-intensive fields. To date, those have included neuroscience and fibrosis. Moving forward, that may also include additional collaborations in areas such as cardiovascular and metabolic disorders, infectious diseases, and more. Each of our recent collaborations that we've announced have come with significant non-dilutive capital in terms of our upfront payments. But in addition to these significant technology access fees, our partners bring far more to the table. In particular, they bring partner knowledge or domain expertise about these complex areas of biology. In addition, they may also bring proprietary data sets or proprietary compound libraries that help expand the breadth of chemical diversity that we can profile on our platform. And thirdly, but perhaps most importantly, is that these partners bring a like-mindedness about industrializing drug discovery and moving beyond traditional approaches. So we've had the pleasure of announcing two of the largest biopharma, biotech collaborations not just in the artificial intelligence-enabled drug discovery field, but in the sector more broadly. And we've done so with two of the most experienced and forward-thinking organizations. The first partnership I'm going to talk about today is our collaboration with Bayer. This is a 5-year exclusive collaboration, including the entirety of the fibrosis disease space. We announced this initial collaboration in September of 2020. At that time, Bayer paid us a $30 million upfront technology access fee, and Leaps by Bayer made a coincident $50 million equity investment to lead our Series D fundraise. In December of 2021, we actually expanded this collaboration to accommodate advancements that we've made in our platform technology, and in particular, a lot of the mapping and navigating tools that Lina and Imran spoke about earlier today. Currently, this collaboration anticipates working on more than a dozen projects, where each one of those projects may yield over $100 million in development and commercial milestones, as well as single-digit tiered royalties on eventual product sales. So as I just mentioned, we started with an original paradigm under this collaboration, what we now refer to as brute force approach. And we recently expanded the collaboration to incorporate these mapping and navigating tools, or what we refer loosely to as inferential search. So under a brute force paradigm, which is where this collaboration began, what we've been doing is physically screening small molecule perturbations on our platform directly on top of disease models of relevance to fibrosis, and measuring the degree to which those small molecules rescue that high-dimensional fibrotic phenotype back to a high-dimensional healthy cellular morphology. Compounds that show initial therapeutic efficacy in that assay are prioritized and validated in more downstream, but still mid or high throughput assay systems, as we pursue them in different indications of relevance to fibrosis and ultimately into relevant patient populations. But with the expansion of our collaboration at the end of 2021, we've really been tapping into the power of the Recursion operating system. And in particular, I go back to some of the comments that Imran was making earlier today. Now having profiled over 500,000 small molecules, specifically of relevance to fibrosis, that came from the Bayer proprietary library, alongside our entire whole genome arrayed CRISPR knockout library, relevant soluble factors, and other genetic perturbations that we've been able to engineer on the platform, we can now infer which of those small molecules may, in fact, provide therapeutic benefit in these different fibrosis-relevant patient populations. We can extract those hypotheses from our phenomaps, and we could take those either small molecule predictions or target predictions, the more enriched and focused libraries from those phenomaps, and we can test them in these more disease-relevant assay systems. Both of these approaches have yielded significant results already for the collaboration. I'm excited to announce here for the first time some of the progress that we've been making under this collaboration. At the top of this pipeline, you can see how some of the initial programs from this collaboration have now moved forward quite considerably and are approaching some pretty critical value inflection points in the, as we transition from early discovery into our late discovery paradigm. Coincident with the expansion of the collaboration to use inferential search approaches, just in the last 12 months, you can see how we've already rapidly expanded the number of early biological insights and programs that we've been able to bring into this collaboration on the bottom half of this pipeline. I'm excited to announce that many of these programs are now accelerating forward under the governance of our joint project teams. So a couple of key stats and figures just to recap the progress we've been making specifically in the last 12 months of the collaboration. Since we signed this partnership, we have initiated 8 projects of relevance to fibrosis. 4 of those projects have been seeded in the last 12 months alone. As we and our Bayer partners who are coming along with us on this journey have really adopted this inferential search mindset, we've been able to really accelerate the pace of innovation. In particular, we've conducted over 40 million experiments on our platform. Those experiments have generated over 2 petabytes of biological image data that now resides within this Recursion data universe. That data set encompasses, amongst many other things, over 500,000 small molecule perturbations that came from a Bayer proprietary library, as well as our own internal small molecule compound library, alongside over 100 different fibrosis-relevant genes that we can now mine for early chemical and therapeutic starting points. Given the success that we've had most recently using these mapping and navigating tools, it's exciting to announce that Bayer has agreed that moving into 2023, we will now begin to source 100% of all future programs using this inferential search paradigm. The second collaboration I want to speak about briefly, but importantly, is our transformational partnership with Roche and Genentech. So we announced this collaboration in December of 2021. It is a decade-long collaboration in the fields of neuroscience, as well as a single oncology indication. At the time of the announcement, Roche paid us a $150 million upfront technology access fee. Additional terms of the collaboration include up to or exceeding $500 million in research milestones and data usage options. Additionally, Roche may pay us up to or exceeding $300 million per program for what may be up to 40 programs that result from this collaboration and our work together over the next decade. On top of that, we will also receive mid to high single-digit tiered royalties on any future product sales that come out of this collaboration. I want to pause here because while very large in scale, what these notes actually miss is the great degree of depth and collaboration that we have on the computation side for this partnership, and the level of data sharing that's really unique between our parties and our collaborators that really form the core of this partnership. So in doing this work, Recursion is contributing significant and very large phenomic data sets. Our partners at Roche and Genentech are contributing extensive single-cell perturbation data sets, and both parties are coming together to really co-invest on the machine learning side with machine learning resources and know-how and investments on the data science and computation side as well. The algorithms and outputs that result from that computation work may be exclusively or jointly owned by Recursion and our Roche and Genentech counterparts. So here I really want to emphasize that the work being done in the Roche and Genentech collaboration endeavors to create multimodal maps of human cellular biology that are of relevance to neuroscience and gastrointestinal cancer, which is the single oncology field that we're focusing on. This work begins by, as has been described earlier today, profiling small molecule perturbations as well as genetic perturbations in these multiple different relevant cell contexts. Having experimentally profiled these different perturbations, we then read out changes in cellular state using either an image-based readout or sequencing-based technologies. Both of those data sets are combined in either Recursion proprietary or jointly developed deep learning models and embedded into these phenomaps, or to the extent they include transcriptomic data, transcriptomaps, if you will. And we are prosecuting and mining these maps for novel biological insights, right? Either target hypotheses or small molecule therapeutic starting points that then we can advance into, again, more mid and high throughput confirmation and validation assays that are of relevance to these two different fields of biology. What's important, and I'll just harken back to the comments that Dean, MD, PhD, completed, mentioned earlier, is we iterate on this loop, right? We iterate with respect to the data, we iterate with respect to the algorithms, and if I recall what Dean was saying, we iterate with respect to how we prosecute those experiments that we're constantly learning, right? We're constantly strengthening our algorithms, we're constantly improving and learning this system as we go in collaboration with our Roche and Genentech counterparts. The progress that we have made to date in this collaboration in just 12 short months has been extraordinary. I speak on behalf of Recursion when I say that. I think I speak on behalf of our Roche and Genentech counterparts as well. We're really enthusiastic about the progress that we're making in both of these fields, and while this is just a small sample of the work that's been done over the last 12 months, we hope that we have an opportunity in the future to speak at far more length about where we're going and the vision for this collaboration. So I will conclude my remarks there. They are short. Obviously, I think everyone in this room can appreciate the disclosure limitations that we have around these collaborations. We look forward to the opportunity in the future to continue to update you on the progress that we have made and the exciting advancements that are to come. I know that means one thing coming from me as a Recursionaut to tell you about these collaborations, but it's even more powerful when you hear from our Roche and Genentech counterparts directly. So I will leave you with a quick video that showcases the Recursion scientific teams as well as our Roche and Genentech counterparts talking about their excitement, right? As Ben maybe invoked in us earlier today, their almost childhood excitement about the power of the Recursion operating system and how we can go and tackle these really complex and otherwise potentially intractable diseases. So I hope you enjoy this video, and afterwards I will turn it over to Heather.
We've had a large contingent out from the Roche and Genentech team, our collaborators. We're talking about science, talking about what we're going to do together, applying what we've built here at Recursion, taking lessons learned and combining that together to find medicines for patients.
You know the metaphor about the guy who loses his keys in the park, but he's only looking under the lamppost because that's where the light is? That's the field of neuroscience. We're all looking under the same few lampposts because that's where we know how to look. And what Roche and Genentech are going to do with us in this collaboration is to build flashlights that will enable us to look in new places and hopefully find the keys to these devastating diseases.
Recursion can collect phenotypic image at an unprecedented scale, and that allows machine learning methods to work much more efficiently.
When I saw the progress being made at Recursion, it became apparent very quickly that the joining of these two formidable organizations would really go a long way in setting new insights into areas of very complex biology and intractable diseases of high medical need.
We made a strategic plan in where can we really differentiate. And that was basically going after new technologies, new drug discovery platforms, big data sciences, computational approaches. Basically, we were looking for partners, and we found in Recursion an excellent partner that basically ticked all the boxes.
It's an opportunity for us, in collaboration with Recursion, to really push the limits of how we do drug discovery, to do things that have not really been done before.
We have strong Genentech people. You have like experts here. 1 + 1 is 2, right? This is like really 1 + 1 is maybe 5. And I think that's where the power is here.
Thanks, Matt. That was incredible. I'll share something with all humility. So we show off about our partners, and I have come to learn increasingly our partners show off about us too. We have pushed them on pace. We have pulled them to move at the speed of Recursion, in addition to many other things. So we learn from them, they learn from us. I love that endpoint of 1 + 1 is 5. I often say 3, but now I'm going to challenge myself to think of 5. And so with that, I now have the honor to bring up the one and only Michael Secora, our Chief Financial Officer.
Thank you, Heather. Good afternoon, everyone. I am Michael Secora. I'm Chief Financial Officer here at Recursion. Prior to joining Recursion, scientist by background, after a PhD in physics and mathematics, became an investor in public and private equities, both in the space of technology as well as biotechnology. A number of years ago, I started to follow and invest in technology-enabled drug discovery. And in meeting Chris and meeting Recursion, saw a company that I felt a calling that pulled me away from my investing world and made me want to join the operating world. I'm very happy to be here with you today and talk about Recursion. So as you've heard today, Recursion has been operating from a position of strength and prudent expansion, and that has been able to drive value across our clinical pipeline, our preclinical programs, our partnership opportunities, our platform, and our data. And we have been able to drive that value from a position of financial strength. We're at the end of 2022, we have finished that year with approximately $550 million in cash and cash equivalents, and approximately $275 to $295 million in operating costs and expenses. Now, based on current operations of the company, we project relatively flat operating costs and expenses in the near term. And we also are projecting that for 2022, approximately $30 to $45 million in revenue, with the potential for increased revenue in the near term with sources from potential partnership option exercises, potential additional partnerships, and continued revenue recognition from existing partnerships. So as you've heard today, Recursion has been uniting the worlds of technology and biotechnology in its philosophy, in its capabilities, in its operation, and its value proposition. But we have also been uniting the worlds of technology and biotechnology investing. Since its founding into its Series A, Recursion has been uniting these worlds because the space, I believe, of technology-enabled drug discovery requires the inclusion of these worlds. It requires the mind share of these worlds to have the potential disruption that we believe it can have. Now, what's shown here below is a number of investors who have supported us from our Series A to our present day. And we are fortunate to have a number of these investors here with us today. Of all of you, just want to say a big thank you for your support of us, for your confidence in us, for your willingness to be company builders with us. And in that, we look forward to engaging with you as we together, this broader Recursion community. You're sitting in the seats that we sit at. You've been in this facility that we are able to inhabit each day. We together can push forward this mission together, that mission to decode biology to radically improve lives. So what does it take to make this happen? What does it take to make Recursion happen? Well, we believe it takes a new kind of team, a new kind of culture. We have approximately 500 employees at Recursion, relatively equally split between the life sciences and technology. And right there again, you're seeing that unification where you see it in their employee demographic at Recursion. Now, as you've been here with us, as you've been able to engage with us, as you've heard from our fantastic Recursion leaders, as you've been able to tour our facility, as you've had a chance to experience a bit of what it's like to be a Recursionaut for a day, it is our hope that you've been able to experience some of that culture that we've been talking about, that you've been able to internalize some of those values that you see written around us here, that you've been able to understand some of those founding principles that we believe help drive our value and help enable us to have a positive impact in the communities around us. And that culture to want to unite technology and biotechnology to have that positive impact, that is a choice. That is a deliberate choice that takes effort. And it is also that culture that I believe is one of Recursion's greatest strengths in the advancement of that mission. Now, I'm very happy that you've been able to be here with us today, hear from our fantastic Recursion leaders, been able to engage with our fantastic Recursion leaders. And I believe in them, we have this experience and we have the innovation to continue to lead and define the space of technology-enabled drug discovery. And throughout the rest of today and into the future, I would welcome you always to reach out to us and continue to engage with us together. So as we've walked through today, how we've been driving value through 2022 with respect to our clinical programs, preclinical programs, partnership opportunities, platform and data, what can we watch for from Recursion with respect to upcoming potential milestones in the near and medium term? In the near term, there is the potential for option exercises for partnership programs. We just heard from Matt how we've been operating against our Bayer partnership and our Roche Genentech partnership. There is the potential for option exercises for map building initiatives and data sharing. Also, we just heard about our Roche Genentech partnership, and earlier today, we heard in 2022 how Recursion had produced over 500 billion iPSC-derived neurons. There's the potential for additional partnerships in large intractable areas of biology or technological innovation. Earlier today, Laura had disclosed to us how we disclosed that connection between RBM39 and CDK12 biology. That's finding novel biology that will be of interest, that could be of interest to large pharma partners. And also earlier today, Ben had showcased to us, had revealed to us the RxRx3 data set and the MolRec application. There again, you're seeing a unification of technology and biotechnology at a business model level. We're providing to the public free resources, freemium resources, so the public can get acclimated to how Recursion thinks about drug discovery in the 21st century. As we just heard from Shaf with respect to our clinical trial, our clinical pipeline updates, later this year, you could expect phase 1 clinical trial data for our program in Clostridium difficile colitis. And there's also potential for additional INDs and clinical trial starts, including a phase 1b/2 trial initiation for our program in Axon 1 APC. Lastly, as Recursion operates from this position of strength, there is the potential to consolidate technologies, talent, and assets to accelerate the Recursion OS that you had heard about from Lina and Imran. And it's in that acceleration of that OS that I believe Recursion will continue to define and lead the space of technology-enabled drug discovery. Now, on the medium term, again, as we've talked about our clinical pipeline, there's the potential for multiple POC readouts, where next year, one could see an interim safety analysis for our program in NF2, and one could see top-line data second half of next year for our program in CCM. Furthermore, there is the potential for additional INDs and clinical trial starts. There's the potential for option exercises for partnership programs. There's the potential for option exercises for map building initiatives and data sharing. And there's the potential for additional partnerships. Lastly, as Recursion focuses on building cycles of learning and iteration, we believe Recursion will move its OS more towards an autonomous map building and navigation operation, with digital and microsynthetic chemistry at its core on par with its biology. So as you've heard today how we've been driving value through 2022, and now you've just heard how we are positioning the company to drive value in the near and medium term, I believe all of this together highlights a maturation of the tech bio value proposition, where there are multiple cycles of learning and iteration across biology, chemistry, technology, all of those cycles coming together that I believe enable Recursion to increasingly provide an enterprise-scale, data-driven, full-stack solution for drug discovery, development, and beyond.
All right. Michael, thank you. And so, Chris, I'm going to bring you up, our co-founder, our CEO. Bring us home.
Thanks, Heather. I hope you all enjoyed learning a bit about Recursion today. I'm going to append some of my remarks so we can move straight into the Q&A. But before I do, I want to just say one thing, which is a huge thank you to everybody at Recursion who helped make this day possible. There's a lot of folks who are delivering pipeline programs and partnerships who were also moving chairs around, putting together slides to make sure that all of you could learn a little bit about Recursion. So a huge thanks to all of you for all the work you did, from the communications team to the IT team to the admin team. An incredible effort by those folks, and I hope you felt it today as our guests. With that, why don't we move to Q&A? Wow, I am really bad at microphones, just the sensitivity level there. Questions? Don't be bashful. Yes, Gil. I think we have a microphone coming your way.
Check. Okay. So it seems like you were reaching a certain level of maturation with your tools, which is pretty impressive. But at that point, what becomes more complicated is which questions to ask of your Magic 8-Ball. And maybe the demo you described earlier today is part of that answer.
Great question. Thanks for raising that. This is one of the fundamental challenges of Recursion. We've got 3 trillion relationships in this map, and that number is going to grow substantially. How do we move that into not just the best insights, but how do we actually get to the point where we can automate some of the toil that will come into exploring this map? And I think one of the things that we've really been focused on, you heard a bit about it today from Lina, from Laura, this industrial program generation is a strategy whereby we can have dedicated teams at Recursion who explore large data sets, clinical data sets, papers that are coming out, and they help look in our map for interesting insights. And then we remove the toil of the next 6 or 12 weeks of work to validate those insights by almost entirely automatically moving them to validation through this phenomics platform, through a transcriptomic platform, so that we can then start investing really deeply in the places where there are validated real insights in our map. And our team can spend their time asking the question, do these matter for patients? But what's beyond that is I think that there are large population scale genomic data sets of interest that one could start bringing into our platform.
that could help start guiding us in a more autonomous way to actually hone in on the most exciting insights that have the highest probability of addressing unmet need. That is work we've not begun yet. It's more manual, but I think that's a place Recursion aspires to go so that we can most efficiently pull all of the data out of the map. Great question, thank you. Next, yeah.
Thanks for the time today. So maybe I want to come at the sort of question of what do you move through your process from the other end, which is, you know, the speed and efficiency of clinical prosecution, right? So you're now in the clinic and, you know, encountering some sort of different challenges, right, in terms of enrollment and in terms of things like study design, I imagine. How do you think about how you might sort of automate, both sort of inject insights about clinical executability into how you choose programs up front, but then also ways of automating and industrializing the clinical trial process? And then maybe how do you think about that across, and how might that be different across owned pipeline versus partnered?
Great question, thank you. What I would say is, can I borrow the handheld? Sorry. Feedback is a little strong. Chef, I'm going to have you come up here. But what I'd just say is it's not a place we've really innovated yet. We're trying to build an incredible clinical development team who can safely shepherd these early assets through our pipeline, because one can only innovate in so many places at one time. And Recursion wants to build innovation at every step of the process, but I don't think today is the time for us to be doing that with all the other work we're doing. So we need to make sure that we don't take too many risks in our clinical programs in the early days. But I could imagine in the coming years, as we finish building out our chemistry platform and our translational platforms, we may get to the point where either through our own internal build or perhaps through partnerships with some of the incredible companies in the space. You know, we've been innovating in drug discovery. There have been companies for the last 10 years or more who've been trying to innovate in the clinical development space. There could be great opportunities for us to partner with the Tempuses or others of the world to help drive some of our programs in a more efficient way. Chef, do you want to add anything since you're leading that team?
No, it's a great question. I'll just try and link it back to, you know, we talked a little bit about recursion mindset. And as we talk about sort of more the distal axis of drug development, which is highly regulated, there's still an opportunity to apply the recursion mindset to really industrialize the later stages of drug development. Examples are, for example, you know, when we talk about our interactions with the regulators, right? So what we're doing here in terms of data generation is white paper level discussions with the FDA, right? In terms of what are these industrialized transcriptomics, phenomics, how does that give us confidence in terms of pushing programs closer and quicker to patients, right? We talk about strategic relationships with our focus in oncology, strategic relationships with phase 1 centers such as MD Anderson, Memorial Sloan Kettering, central IRB processes that really decimates the normal review times. So I think there's an ample opportunity in addition to large patient population data sets that we can reverse engineer to the level of clinical trial design stratification, but also fundamentally to more proximal and to looking at how we better query our maps and really mine real-world data in terms of mutational profiles and make more targeted interventions there. So, I mean, I think we watch the space closely. I think there's going to be a lot of movement moving forwards there.
Thank you. All right, next question. I saw a couple, there we go.
Hey, Alex Stranahan from Bank of America. One question I had, you know, your current pipeline is all small molecules. Is that sort of lower hanging fruit for your platform in your view? And is there potential flexibility down the road to shift to biologics as well?
Yeah, it's a great question. I think there certainly are targets that are going to be most amenable to other modalities of treatment. And if we find in our map those targets, we will not hesitate to partner with some of the leaders in a variety of other modalities to help drive those programs forward. To the earlier question, we can only innovate in so many places at once. And this is a very innovative place. And I think what we need to demonstrate to all of you, to the market, to our partners, is the ability to drive programs all the way through proof of concept with one modality, which is small molecules. We've chosen that for a variety of reasons. If we're able to demonstrate those proof of concepts, I think we will very aggressively move in a horizontal direction
to take all of the learnings, all the mindsets, all of the data, to then expand into other modalities, which may be the ones we're talking about today, or they could be different in 3 or 4 years. But I think there will be new learning that's required, new data sets that are required, new algorithms that are required. But I actually think that horizontal expansion into those other modalities is going to be less of an innovation for Recursion than taking our programs all the way through into the later stage clinical portions of the pipeline. Thank you. Great question. Yeah, next.
Thanks, Chris. Matt talked about the like-mindedness with Bayer and Roche Genentech, and I was wondering, I guess, 2 things. How would you sort of describe the state of like-mindedness away from those two in the big pharma community today? And then secondly, how does Recursion go about actually vetting for it as you seek out partners?
Matt, why don't you join me? I'm going to give a brief answer, and it's going to be lawyerly, and I'm going to say it depends. There's actually a broad spectrum of perspectives in this space, and I would give a lot of credit to some of the large pharma companies who, they are moving very big ships with incredible momentum and a lot of focus on late-stage programs that are on the cusp of delivering to patients. And I would say over the last 3 or 4 years, incredible nimbleness for such big entities to start really thinking about this space. Now, certainly companies like Roche Genentech, putting Aviv Regev in charge of research at Genentech, that's a huge signal to this field. You have really the first computational biologist leading one of the largest research entities. And I think that is a harbinger of what's to come, but some of the companies aren't there yet. So I'll turn the rest of the eloquent answer over to Matt.
No, I'll just add to it. I'm sorry, show who asked the question, because I had my back facing to you. Thanks. I think there is a quickly growing group within large pharma who appreciate and who are on board with the vision. What I find and what we find often in our conversations is there remains at times an underestimation about how hard it is to build scaled, reliable, relatable data sets that Lina and Imran were talking about. And sometimes that underestimation leads some pharma partners to believe that they can create what we're creating. And I will say some of the easiest conversations that Shafique and I have is when those pharma partners have attempted to go and build what we've created, only to come back and have a conversation with us. That's also why, as you all have experienced today, it is so important to come here to see, to touch, to feel, to interact with the data sets, the robotic and automation setups that we've built. Because I think it is underappreciated that the amount of capital, the amount of time, and frankly, the number of failures before you can really start learning that's required to build the systems that we're building. So the vision, I feel like most people have a crystallized vision for how they're going to use artificial intelligence. And I think increasingly, as more people are coming to the realization that it's companies like Recursion and it's companies who are building very intentionally those proprietary data sets who are going to be successful. Yeah.
Thanks, Matt. Great question. Yes?
The presentation today, Chris. Just one on pipeline strategy. Moving forward, I guess you could say medium to long term, right? Should we expect, primarily NCEs in the pipeline, or are you guys still possibly considering, in licensing, external compounds as you have in the past?
Yeah, so we will certainly be opportunistic. If there's a medicine that is closer to the clinic with great data behind it and our map gives us an insight, we will not be shy about trying to in-license those medicines. One of the things that I think I got most wrong in the early days of Recursion, is that I believed that all incentives would align when it came to in-licensing these assets from large partners. And though many of them, I think, emotionally are aligned, there's a variety of sort of perverse incentives in our industry that are no one individual's fault, they're just the way the industry has grown, that makes it really challenging in some circumstances to bring those medicines in. But when we find the opportunity and the data supports it, we will be aggressive to bring those in. That said, you're absolutely right. I think the vast majority of programs coming through our pipeline are going to be new chemical entities. And I think one of the reasons you're seeing us invest so deeply in small molecule chemistry, the predictive ADME program that you saw at the top of the stairs, some of the work that Imran shared and Lina shared around the digital chemistry tools we're building, even our own ambition to continue growing with automated microsynthesis, those are in service of our belief that the vast majority of the programs will be NCEs. Thank you. All right. Oh, we don't have the microphone on yet. Thanks.
Hi, okay, just thinking about Rec 4881 and then how you're taking it into both FAP and then Axon 1 APC. I don't think this is the last time that your platform is going to be able to identify more than one indication that a chemical compound could be attractive in. So just thinking about how you decide which indications to take forward and how you prioritize those.
Great, I'm going to invite Shaf up to answer that one.
That's a great question. I think during my overview of the clinical programs, I mentioned a couple of times about the power of inference in terms of indication expansion, talked a little bit about life cycle expansion, both in the setting of new insights that we're getting that are being generated from the platform. I think then really the case becomes of rapid diligence in terms of what is the size of the unmet medical need here? Today, for example, in cancer, we have 2,500 clinical trials that are ongoing. By God, this is going to change the landscape of cancer therapy in the next 3 to 5 years, right? Where we think today unmet medical need sits, that landscape will look very different in 3 to 5 years. So we have to remain true to our mission in terms of the insights we're generating. On one hand, we'll never leave any insight that has potential to impact the patient's journey without oxygen in terms of moving forwards. But at the same time, I think we have to be pragmatic in terms of, where do we see rational combination therapies, a lot of combination trials ongoing at the moment, where do we see areas continuing to pose unmet medical need or areas that will bring up emergent medical need in terms of how we sort of, prioritize these programs. I think at the moment, fair to say, we have a very nimble strategy, and that's why the common slide that Matt and I shared that goes across corporate development and clinical development in terms of how we think about value. I think we have to be very pragmatic in terms of what can we execute as Recursion. A lot of the cancer therapies, a lot of the targets that will emerge will probably be combination plays, in which case we will leverage our corporate development, business development access to bring in partners who have those portfolio pipeline molecules to start, combination trials sooner. So I think what you'll see is a very dynamic picture moving forwards in terms of, how we create value, how we execute programs, either as Recursion de novo or where we bring partnerships as well on that.
Thanks, Shaf. All right, we've got time for a couple more. All right, thank you everybody. With that, I'm going to invite up our final speaker of the day, Zavain, who led our Series A investment. Big day for you as well.
Thanks, man.
Take it home.
Hey, I suspect Chris invited me to give this final talk here because we offered to pay for dinner tonight, which everyone is invited to. Cheers, yeah, okay. Like Marty, I think the one thing I am an expert in is myself, although 12 months into launching my own company now, I think my two co-founders likely know me better. Much to Dean's chagrin, I graduated from Stanford, undergrad and graduate school in computer science. Chris asked me to give the context when I first led the Series A from Lux into Recursion. I'd spent the 4 years prior advising first the Houston Rockets and then the 76ers as any 5'9", somewhat athletic but not very athletic brown boy would want to do. And was coming in right during the kind of massive data-driven digitization of sports. If anyone has watched or read Michael Lewis's Moneyball, what happened with baseball is what was happening with basketball. I learned two things. To be an investor or an executive, there's two ways to win. You can play the same game that everybody is playing, but really take a bet that you are just the best at it, or you can realize that the rules on the field were changing, and you could make your own strategic changes to the changing rules on the field. As what happened with baseball and as what happened with basketball, the rules were changing. If you follow the league today, and I don't need to go too much into a black hole about this, but if you follow the league today, people shoot more threes. The way we draft players is entirely different. You don't shoot long twos, you want to get to the line, so on and so on. And when I first met Chris Gibson, he was one of the first entrepreneurs who really realized that the rules in biotech, that the confluence between molecular tools and automation and software and ML were changing. And so that led to the investment in Recursion. I think Chris says this publicly, every private financing he ever had, he only had one term sheet on the table. So there was literal existential angst the whole way through the company's life cycle. When we led the Series A from Lux, my prior firm, into Recursion, the firm was candidly stepping back from biotech. We had divested our biotech portfolio. We were considered not to be super great at it. And candidly, it was largely driven by the success of Chris and the entire management team you saw today and Recursion and that vision that gave Lux confidence to really double down into the ecosystem. When we made that investment from Lux, and this is hopefully to give the financial players here a sense of proxy and the analysts here, when we made that investment from Lux, the firm had under 500 million in management. And today that firm, Lux, has over 4 billion and has publicly committed to invest over 1 billion exactly into this ilk of company. So that's a little bit of I think the trajectory of at least one firm's dive into this space. You also saw two other board members, Dean and Marty, both far more eloquent and credentialed than myself, speak. I think you saw the two poles. You saw in Dean a traditional molecular biologist, and you saw in Marty someone who really ascribed, if you caught him yesterday, to a potential singularity from AI in the next decade. I quite frankly think Marty was potentially underselling. He and Dean spoke about Irum's law, the realization that this industry, this subsector of the GDP, this largest subsector of the GDP, 20% of the GDP, is the only modern subsector of the industry that doesn't have increasing economies of scale and or productivity, right? It's not just linear, it's not just 1 grain of rice, 2 grains of rice, 3 grains of rice, but it's actually 1 grain of rice, 2 grains of rice, 2.5 grains of rice, 2.23 grains of rice, so on and so forth. That's what this industry is up against. And our realization is, quite frankly, not only on the ML and automation and software side, which does draw a lot of the headlines and the attention, but the realization that every assay of merit, when you all walked across in those demos today, does follow Moore's law, exponential curves in cost and scale and fidelity and reproducibility. And the modern lab today is the largest generator of data after only the hyperscalers. If you go and talk to any of the execs at Microsoft, Snowflake, Amazon, Google, so on and so forth, they are all, as my partner Nan would say, following the petabytes, and they all have their eyes and ears exactly here and are looking at the Recursions of the world. I'll come back to Recursion in a second, but I don't think this is super theoretical anymore. If you go to any of the leading molecular labs, not just Dean's lab at University of Utah, but maybe you can go to Stanford, maybe you can go to Harvard, Yale, so on and so forth, all of them today are practicing distributed TensorFlow in ways that maybe 5 years ago were very, very, very rare, and in 10 years ago were absolutely null. Conversely, if you go to any of the leading cloud computational companies, the Googles, the OpenAIs, the DeepMinds, the Meta AIs, the D.E. Shaw researchers, the so on and so forths of the world, they're all one by one individually building their own wet bench capabilities. They're attacking with increasing traction. It's not just AlphaFold, it's many other problems, but they're attacking with increasing traction biology and chemistry's most vexing problems. Recursion is at the forefront, and it's the leader in this space, and it's really the only company that's attacking it in, I think, a sophisticated manner. But the question of, is this possible? It's no longer really a theoretical exercise. It's very much so, the existence proofs are here. For anybody, I think, with a discerning eye, you can look and you can see what's possible. Capital markets, as you all know, are the lagging indicator. And capital markets today are still very much so reflective of biology and technology from 2 or 3 decades ago. That's not surprising. It always has been the case. And I think the analogy that's easiest, at least for me to think about as a millennial, is the cloud. If you look at kind of the first cohort of cloud companies that went public in 03, 04, 05, and 06, for the first 2 or 3 or 4 years, they were largely valued as on-prem software companies. The KPIs, the waypoints, the goalposts that management teams and boards set up those companies against were different than how analysts reviewed them, and were different than how investors invested in them. It took a while for there to be a standardization of vocabulary, and I think that's where we are in this moment in time with tech-enabled bio and biotech. There's a lot of fear and anxiety, certainly especially in this bear market. I will use, with a little bit of humility, our own fund as hopefully a positive breadcrumb for everybody here. If you remove rxrx.ai from your web browser and go to dimensioncap.com, we just launched today. We launched 12 months ago, where we left our prior firms 12 months ago to raise $300 million, well before all of the buzzword bingo of inflation rates, base rates, Ukraine, Russia, so on and so forth, were on the tip of everyone's tongue. In an environment where it was so easy for every investor to say no, we not only hit 300, but we hit our hard cap of 350. And that's not, as you can see, because I'm a great public speaker, but I think it's because the leading allocators of the world, our investors who represent the top universities, endowments, pensions, foundations on the planet, were keen and eager to deploy capital against this. Again, going back to thinking about 2 ways to win, there's one way to win where you continue to play the same game and you just take a bet that you are better than everybody else. But I think as an investor, there's another way to win where you realize the rules, the primitives are changing, and you take that bet. And so that's certainly what we decided to do. I think as an operator and as an entrepreneur, that's what Chris has decided to do. But I encourage others to think about taking that bet with us, because I think fundamentally, the primitives are all there, and this is no longer a theoretical exercise. You can debate me at dinner, but that's all I had. Thank you.
Zav, thank you so much. Okay, all right, everybody. We've got some snacks and drinks in the lounge just around the corner here. I'll see you there. Thank you very much. Thanks to all our speakers today.